I want you to your boss. And you can give your remarks of care.
Okay.
You can give it.
Okay. Bismillahirrahmanirrahim. Assalamu alaikum and good morning and good day, ladies and gentlemen. I'm Hazima Wazan, Program Manager Comstech. I welcome you all to Comstech Azerbaijan workshop on nature-based medicinal and cosmetic products. Ladies and gentlemen, this workshop will be delivered by eminent scientists from Azerbaijan and Pakistan working in the area of natural product chemistry. So without waiting any further, we will begin today with the recitation from Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Jazakallah. Ladies and gentlemen, I would now like to request His Excellency Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General Comstech, for his welcome address.
Bismillah ibn Shaitan Rajim. Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, ladies and gentlemen. Excellency, my dear brother, Hazir Nadir Farhador, the ambassador of the Republic of Azerbaijan, very close to our heart. Excellency, the ambassadors of a very strong supporter of OIC Comstech. And we also have Excellency, the Dean of Ambassadors in Islamabad, and he has been such a source of inspiration. Distinguished members of Azerbaijani delegation, particularly Professor Dr. Sayara, my dear sister, who is leading this delegation of 8 scientists from Azerbaijan. Valued participants, guests, distinguished speakers, ladies and gentlemen, it is indeed a very important occasion. We've been planning this event since long, and finally the delegation is here. There was a restructuring in Azerbaijan, the Institute of Botany, which is a very prestigious institution, was originally part of the Academy of Sciences of Azerbaijan, which was recently has moved into Ministry of Science and Technology of the Republic of Azerbaijan. And that restructuring has really stopped us to have the honor of receiving this distinguished delegation. But now they are with us, and it opens new avenues. Ladies and gentlemen, nature is particularly important for us. Think of a world 100 years ago when there was no synthetic medicine, when there was no biological, no vaccines, no injections, no immunization, nothing like it. And nature has kept us alive. Humanity survived. Even our forefathers used to have better and longer life than us. Isn't it, brother? So nature has been the savior of humanity in the most turbulent time of the history. And nature is constantly striving. During COVID-19, when we were looking for vaccine, and by the way, now there are recent research that messenger RNA vaccine, which we all have injected very happily, now proved to be unsafe. After dispensing it to 7 billion people, they found it that it is not scientifically validated. During that turbulent time of COVID-19, we were looking for natural products. And this event has a particular significance because here we are trying to connect ethnobotany, traditional medicine with science. In this day and age, without scientific evidence, it's not possible that we can promote and propagate natural medicines or anything else, including natural cosmetics. And this is exactly what my dear sister, Dr. Sayara, has been doing. She's a leading expert in the field of ethnobotany. And her work is considered to be a class of its own, that she has been compiling Red Book, which is the most endangered and vulnerable plants of Azerbaijan. She has also worked on a lot of taxonomy and ethnobotany of plants. Most recently, after the liberation of Karabakh, she has compiled this fascinating treatise of the plants of Karabakh, which was in between destroyed during the occupation. So she has been an iconic personality. I must say that she has received a very prestigious award during COP19, the Conference of Parties in Azerbaijan, COP29, sorry, and she has received this. But I'd like to tell you that why it is important to link science with natural medicine. There are two important components. Number one, that nature remains a very important source of medicine for us. You see, even 40% of the medicine, if you go to the pharmacy shelf today, are of natural origin. According to the World Health Organization, 70% of the world population rely on natural medicine for their primary healthcare needs. It's also important for us that now there is a new paradigm, that there is a need of disease prevention and health promotion, so-called science of wellness. And this is where natural products play an extremely important role. Now, I'll give you some statistics of what is happening. Our World Health Organization has now drafted a global traditional medicine strategy, 25 to year 2034. And this binds all member states to integrate natural medicine as part of their healthcare system. Seamless integration of traditional medicine in the healthcare system. And on top of it, we have in the Muslim world, Islamic medicine, prophetic medicine. Our prophet has actually mentioned many things which now modern science tells us that they are the best things which are available for keeping the health of human at the wellness level. Prophetic medicine, Unani system of medicine, Persian system of medicine, and others are extremely important. And now, if I tell you that many countries of the world are heavily investing into research of traditional medicine, TCM, for instance, traditional Chinese medicine, there are in TCM there are 24 universities, and there are 600 TCM hospitals, 1.15 million bed hospitals. So this is important that we move forward and do science in it. Well, I'll give you global business. If you're not convinced that health is through natural medicine would be the only reason of investing into research, then I'll tell you about the global business of botanicals. And yesterday we were discussing with Dr. Sayara and Dr. Dilzara, natural cosmetics. They are expert in it, And you can see their beauty is largely because they've been using natural medicine. Natural cosmetics, 41.65 million USD, will reach to 103 million USD by year 2034. Aromatherapy products, 1.5 billion USD, and will reach to 10 billion USD by year 2032. Fragrances, essence, perfumes, 55.53 billion US dollar will reach to 88 billion US dollar in 2033. Botanical drugs, 233 billion US dollar and will reach to 434 billion dollars by year 2032. Health food, 900 billion US dollar in 2024, and it will reach to $1.6 trillion by 2030. Cannabis, cannabis, which are now natural cannabis, which is used for both recreational and medicinal purposes, is 1.1 billion US dollar global business. Ginseng, which is very common in the Far Eastern world, $1.28 billion. And if I tell you that even the very specific cultural oath, which we in the Muslim world use, is business of 1.89 billion US dollars. So you're talking about money of a magnitude which is much larger than new technologies, I'll tell you. You talk about creating money through technology, but the fact is that this is the money which is right there in front of you in which you can contribute. But what are the major sources of medicinal aromatic plants from where these plant goes? South and Southeast Asia, Central Asia, Africa, and South America. Major botanical companies such as IFF Australia, Gavaudian Swiss, Simrise German, Calsec USA, and Marinbauer Germany. They are the companies who add value, so they get raw material from us and they add value. I'll give you the example of Body Shop. $503 million US dollar Body Shop, one company. GNC, 227 million US dollar, and Christian Dior, 84.7 million euros. So this is what the global business tells us. Now, Azerbaijan, with its very unique geographical Eurasian plate, two different continents meeting together, has a very unique flora. And that unique flora enable them to actually use their natural sources. It has 8 climatic zones, from desert all the way to high mountains, and Azerbaijani flora over 4,600 plants is one of the richest in that part of the world because it has been preserved and quite. So with this, we would really like that we work together with Azerbaijan. Assalamu alaikum, brother. Thank you very much for coming. That we work together with them, and we have agreed on exchange of scientists. We agreed that we would have industrial linkages. We agreed that we would work on institutional linkages with them. And we have also agreed that with our premier research institution in Karachi, which is International Center for Chemical and Biological Sciences, one of the finest center which work along with different fields in the field of natural products since last 60 years, we will build these stronger collaboration built upon what we have been able to do in last 40 or 55 years. With this, I'd like to thank you all. It has been a great pleasure of having you. It has been a great pleasure of hosting Azerbaijan delegation. It is a watershed moment. It is a time that we celebrate the diversity. It is the time we celebrate the unity of Islam. And it is the time we should understand that comstech, being a premier organization with tremendous work, is the center for scientific-based socio-economic development. Thank you very much. Thank you.
Thank you very much, sir, for the welcome remarks and also highlighting the significance of nature-based industry. With this, we will thank you very much, Excellencies, for joining. With this, we will move on to the introduction of the workshop. This will be delivered by Professor Dr. Atiyatul Wahab, who is a full-time professor at Dr. Panjwani Center for Molecular Medicine and Drug Discovery, International Center for Chemical and Biological Sciences, University of Karachi. Please welcome Dr. Atiya.
Bismillahirrahmanirrahim. Assalamu alaikum and good morning, ladies and gentlemen. His Excellency Ambassador of Azerbaijan, His Excellency Ambassador of Somalia, His Excellency Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General Comstech, Distinguished Professor Dr. Sayara, distinguished delegates from Azerbaijan, esteemed government officials, faculty members, colleagues, and very dear participants. I was given the responsibility to introduce about the workshop, as we all know that the growing global demand for safe, effective, and sustainable healthcare and beauty solutions has reinforced the importance of natural products as a source of therapeutic agents and cosmetic formulations. From traditional remedies to modern innovations, nature remains an invaluable reservoir of bioactive compounds with vast medicinal and cosmetic potential. In this context, OIC Comstech, in collaboration with Azerbaijan, is pleased to organize the workshop on nature-based medicinal and cosmetic products. A distinguished delegation from Azerbaijan is visiting Pakistan to participate in this event, symbolizing the strong ties of scientific cooperation and cultural exchange between the two countries. Their presence highlights Azerbaijan's rich heritage of traditional medicine, biodiversity, and modern advancement in natural product research, which will add depth and diversity to the workshop discussion. The workshop aims to bring together expert researchers and industry professionals to deliberate on the chemical and biological diversity of natural resources, innovative approaches for the development of herbal medicine, nutraceuticals, and cosmetic products, integrating traditional knowledge with modern science and technology, ensuring quality assurance, safety, and regulatory compliance for international markets, strengthening regional and international collaboration for research and commercialization. Through this initiative, OIC Comstech and Azerbaijan seek to promote knowledge sharing, build capacity, and encourage innovation in medicinal and cosmetic sectors. The workshop also reflects the commitment of both countries for advancing health, well-being, and economic growth across the OIC region through the responsible use of nature's resources. Thank you.
Thank you very much, Professor Atia, for introducing us to the workshop. With this, we will now request Professor Dr. Sayara, who is Director General, Institute of Botany, under Ministry of Science and Education, Republic of Azerbaijan, for her remarks. Her remarks will be in her native language, and we'll request Dr. Emil to translate.
Salam alaikum. Dear organizers, fellow colleagues, dear friends. It is my great pleasure to convey my sense of gratitude to you. It's a great honor for me to participate in this prestigious seminar titled Newcastle in Botany, Pakistan, Azerbaijan, bilateral seminar on national medicinal and cosmetic health in Pakistan. I would like to express my deep gratitude to the comstech General Coordinator, Professor Mohammed Iqbal Chaudhary, and all the staff for organizing this event. I am sure that this seminar will make a significant contribution to further strengthen scientific cooperation between Pakistan and Azerbaijan and to the exchange of new knowledge, ideas, and experiences in the field of national medicinal and cosmetic. Our common goal is to study in more depth this rich resource that nature has given us and to combine them with the achievements of science in the way that we serve human health. I wish all participants successful discussions, loyal collaboration, and fruitful results. Thank you very much, Professor Iqbal Chaudhary. Thank you very much, Hanım Hazima and Hanım Atiye. Hörmətli səfirimiz buradadır. Fəzəl müəllimi də salamlayıram xanımla və onları görməkdən çox şadam.
I also would like to greet the Excellency, the Ambassador of Azerbaijan, and his wife. Thank you very much.
Thank you very much, Madam, for your remarks and for your appreciation. Now I'll request our chief guest, His Excellency, Mr. Khazar Nader Farhadov, Ambassador, Embassy of Republic of Azerbaijan, for his remarks. Please welcome.
Salam alaikum, dear my brothers and sisters, excellencies, our great brother and friend, Dr. Chaudhary Saab, our delegation, Madam Sayyara, Dr. Sayyara Ibadullayeva. Of course, it is not so easy for diplomats to talk on this topic, but anyway, I try my best because it's very important from many points. As it said, as a scientific event and also making the new bridge between our two brotherly nations, and also not only between Pakistan and Azerbaijan, also for among the all Muslim brothers, member of the OIC countries. As you know, Azerbaijan-Pakistan relations is one of the unique and exemplary relations, I can say, for the countries who would like to serve for the peace, security, and development, not only for their own region, but also overall in the world. I think in for that purpose, OIC and OIC specialized institutes, especially the comstech specialist, and plays a very important role because comstech for science, technology, and education, which is the future of all the world, including our Muslim world also. That's why we highly appreciate the successful, activities of the comstech under the leadership of my brother, Dr. Iqbal Chaudhry Saab. I think this today, which we witnessed, and today workshop we will witness also, this research and the contribution and results that Azerbaijan scientists in this field under leadership of the Madam Dr. Sayar Ibadullah are doing. It is very much plausible and very much, success not only for Azerbaijan, for all of our region and also for OIC countries, OIC family. And these days, health security became very important because this, with the growing population and the, challenging economic, ecological situation in the world, directly and indirectly impact this health situation in the world, not only human being, but all the ecosystem. That's why going back to our roots to use this more, let's say, natural way of handling these things and overcoming this, health issues or related to these things, another development is very important. And another good sign in this regard, in this development, is the liberation of our Karabakh. We call it Jannah of the Earth area of Azerbaijan, as every country has similar part of the regions, but Karabakh also one of them. As my brother, Dr. Muhammad Iqbal Chaudhry mentioned here, uniqueness of Azerbaijan's ecosystem being in the crossroad of the west and east, and ecological impacts for both sides as a nature also bring the unique, kind of, let's say, characteristics of the region. Just one example for you, I would like to bring to your attention. We have special plant which we call Kharibulbil. It comes from the family of orchids, only, mainly growing in the Karabakh region. It is very unique, actually, in unexplainable things that it's only, growing in the Karabakh region. When we liberated Karabakh, it automatically became the symbol of our victory because we couldn't see these plants almost more than 30 years. We couldn't smell, we couldn't, enjoy its beauty, but alhamdulillah, after, special, let's say, preparation, solidarity, and in my country, under leadership of my president, and also the solidarity in the Muslim Ummah, in the morally and political support from brother countries like Pakistan, Turkey, and being in the just position and based on the United Nations Security resolutions, at the end we achieved, and I think it showed the world that justice achievable, but if you are in the right position, if you are in the right solidarity and unity with those countries, with those powers really interested in stability, peace and prosperity, not in the world, but not in the country also, but in also all the world around us. Of course, I am not a specialist, but as my own, general knowledge, I can say that natural medicine history in Azerbaijan very deep, very rich. From childhood, even we heard a lot about our, magic, impacts, effects of the curing effect of the plants of Azerbaijan. Even myself, when I was a child, sometimes we can, cure something with synthetic medicine, but we applied directly to national medicine, and it immediately, trade and cure this all disease what we, face it. From my own, life experience from childhood, I several times, met the situation, and in the positive, understanding, we positively affected from this treatment also. That's why sometimes when I'm talking about, this our breeds, historical breeds between Pakistani peoples and Azerbaijanis. I'm always repeating and bringing an example, Multani Saray. Multani Saray is the special structure built as a caravanserai form, which means in the translation, hotel type structure, which, hosted guests from different countries. But this structure specially built from Multani traders, and it was built 600 years ago. I always ask a question to me, how it can Baku became so popular? I mean, in the subcontinent, why the people took this almost 2,000 kilometer travel to reach the Baku from Multan. It is not close to each other cities. It is not even the same region. But sometimes they came to my mind, maybe the oil, because oil in Azerbaijan came surface because of the altitude of Baku city, which is the low beyond the world ocean level. And oil came surface itself. You didn't even at that time intermediates to go for industrial way to extract the oil. But oil itself also, we know, have its own. It is also, not synthetic. It's first form, it is organic form, it's natural form. It's also used for, at least even today, they are using for example, naphthalene oil, very, popular in Azerbaijan and around the whole, region surrounding Azerbaijan for the treatment of this many diseases, including skin disease, joint disease, maybe also one of the, another reason as Kodek, Multan, and, Baku, and is also made one also its contribution for the exchange between our two peoples, and at the end, I think, for the brothership and friendship, which we are right now reviving this. That's why we can, sing the mission of Comstech today, and Madam Dr. Sayari Baduleva for the reviving this kind of, cooperation and bridge, not only between Pakistan and Azerbaijan, but under Comstech chamber umbrella for all the Muslim world. If you just go for the research of the not from only eastern side of the world, if you go the western side research of the at that time Westerns, the travelers, they always so many time mentioned about the special, strengths of the Azerbaijan's herbal, fauna for the flora and fauna for the, this in the medicinal, treatment. And of course, I will not go more in deep because it is very difficult for me. Of course, it is good. It is a great privilege for me to be here with you, at least as a diplomatically, do some my own contribution and personal capacity also, and as an ambassador of the Azerbaijan in the brotherly country. I will not take your time much more. I just tried to explain the importance. It is not only scientific, topic, important topic, but also, from the bringing two brothers countries closer. If it is the health issue, it is the issue regarding the health. I think it is one of the important points that two brotherly countries and all the Muslim Ummah can grow solidified. And it is not only for today's, let's say, today's problem solving, but it is future oriented because it's aiming, if we can eliminate and we can focus effectively in the natural medicine, I think we can build also our healthy and safer future of all our young generation. Thank you very much for your attention. Thank you.
Thank you very much, Excellency, for your remarks and for your valuable input. With this, ladies and gentlemen, as we are nearing to the closing of this inaugural ceremony of this event, on behalf of Comstech, it is my great honor and pleasure to extend heartfelt vote of thanks. First and foremost, His Excellency, our Chief Guest, Mr. Khazar Nadir Fakhadov, Ambassador of Republic of Azerbaijan, for his gracious presence and valuable time here. Thank you, Excellency. We are especially indebted to our partners and guests, of course, from Institute of Botany, Ministry of Science and Education, Republic of Azerbaijan, led by Professor Dr. Sayara. Thank you, Madam, for your time and for being here with us. Of course, our esteemed colleagues who are accompanying her for this visit, we are also thankful for your valuable contribution and scholarly presentation that you will be giving here today. Our profound thanks goes to Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General Comstech, for his inspiring leadership and vision, which has made this workshop and many other collaborative scientific endeavors possible here at Comstech. Our sincere appreciation also goes to distinguished speakers from Pakistan who will be delivering talks on diverse aspects of natural product sciences and their application. And these experts are Professor Dr. Raza Shah, Director ICCBS, Professor Dr. Atyatul Wahab, Professor PCMD, Professor Dr. Asma Dar, joining us from Hamdard University, and Professor Dr. Jamshed Iqbal joining us from Comsats University, Islamabad. I would also especially like to thank ambassadors and the representatives of OIC member states who joined us for the inaugural here, including some eminent guests from like President of EcoSF, Eco Science Foundation, for his presence. I would also like to thank member Pakistan Science Foundation for his presence here today. Ladies and gentlemen, with this, I would also like to appreciate our young researchers and participants who are present here on site and online, who would be engaging in the technical talk and thought-provoking questions and answers, enriching the discussion of this workshop further. A special thanks is due to Professor Dr. Atiyatul Wahab and Professor Dr. Delzara for being the technical focal persons for this workshop, providing the context and the materials for this workshop. A word of gratitude is also due to the organizing team of Comstech and the supporting staff who worked tirelessly to ensure smooth execution of this hybrid event. Ladies and gentlemen, I would also like to thank my colleague Mr. Murtaza Noor for coordinating the media and the participation of Comstech Consortium of Excellence participation in this event. It is with this that it is hoped that this workshop will be a meaningful platform to exchange knowledge, build networks, and explore opportunities for future collaboration between Pakistan and Azerbaijan. And paving to research projects, capacity building initiatives, and greater utilization of nature-based resources for health and economic development. Thank you, and we look forward to welcome you again at Comstech in our future events. With this, with permission of Professor Dr. Iqbal, I would request him to present souvenir and shield to our chief guest, His Excellency Ambassador of Azerbaijan, or maybe downstairs, or maybe here is okay, to Her Excellency Ambassador of Republic of Azerbaijan. Thank you very much, sir. Now I would request His Excellency Professor Iqbal uh, and His Excellency Ambassador of Republic of Azerbaijan to award souvenir and shield to Professor Dr. Seara, DG Institute of Botany. Ladies and gentlemen, thank you very much. And we will now move on to a group photo, which will be held outside the auditorium. It will be followed by tea, and the tea break will be of 15 minutes, and then we will gather here at 10:35. Please join us for the group photo. Ladies and gentlemen, welcome back. We will begin the technical session of our workshop. And this session will be chaired by Professor Dr. Mudassir Asrar. Dr. Mudassir Asrar is an eminent scientist in the field of natural product chemistry, and she is a professor emeritus at University of Balochistan, Pakistan. The first talk that we are beginning with is by Professor Dr. Seara, and the title of her talk is Folk Medicine and Ethno-Pharmabotany. And in this presentation, she will also be highlighting current problems and challenges of study of useful plants of Azerbaijan flora. Professor Seara, please welcome Professor Dr. Seara.
Selamun Aleyküm. Ben çıkışımı Azerbaycan dilinde edeceğim fikirlerimi tam çaktırmak için.
I will be speaking Azerbaijani in order to express
Benim çıxışım Azərbaycanın dərman bitkiləri, xalq təbabəti və etnofarmabotanikadır. Öncə Azərbaycan haqqında qısa məlumat verim. Azərbaycan 11 milyon əhalisi olan bir ölkədir. Onun 58% dağlardır, 42% düzənliklər, 27% 1000 metr dəniz səviyyəsindən yuxarıdır, 18% isə 26 metr dəniz səviyyəsindən aşağıdır.
I would like to give a short brief description of Azerbaijan. Azerbaijan has a 11 million population and 58% of it consists of mountains, and 27% of them are more than 1,000 meters elevation, and 18% of them are below the sea level.
Azərbaycanda deməli kulturlu florada daxil olmağına mədəni florada 5000-ə qədər bitki var. Bu 200 fəsilədə 1100 cinslər birləşmişdir.
In Azerbaijan, there is a totally approximately 5,000 plant species. They all they join together in 200 families.
İlk öncə istəyirəm Azərbaycanda etnobiyologiyanın tarixindən danışım.
I would also like to give a short speech about the little history about ethnobiology.
Etnobiyologiya, etnobotanika və xalq təbabəti Azərbaycanda orta əsrlərdən geniş yayılmışdır.
Since the Middle Ages, mid-centuries, the ethnobotany and traditional medicine have been widespread in Azerbaijan as part of ethnobiology.
Qədim əlyazmalarda bitkilərin istifadəsi haqqında çoxsaylı kitablar, əsərlər günümüzə qədər gəlib çatmışdır.
Too many books as a form of old scriptures have came to our time.
Amma bu ənənə niyə qədər davam edir? Gördüyünüz şəkillər müasir dövrdə Azərbaycan xalq təbabəti dərman bitkilərinin toplanılması, saxlanılması və satışı ilə bağlıdır ki, bundan indi də Azərbaycan xalqı istifadə edir geniş.
And this tradition also continues till now, as the collection of the medicinal plants, their storage, their sale are also currently available in Azerbaijan right now.
Azərbaycanın yabanı florasında qeyd edelim, 5000, onun 4547 növü yabanı floradadır və bunun 1800 növü dərman bitkisidir, 1000 növü aromatik bitkilərdir, 350 növ bitki təbiətdə azalan faydalı bitkilərdir ki, onlar artıq bədəniləşdiriblər, becərilirlər. Yem qrupu bitki 2400-dən çoxdur, bunların da tərkibi zülalla zəngindir. Texniki bitkilər, yəni boyaq, rəngləyici, ip hazırlamaq və müəyyən tapda şalban məsələlər 350 növdür və yabanı tərəvəzlər də 800 növdür.
In terms of wild flora of Azerbaijan, there are totally 4,547 species, and 1,800 of them are medicinal plants, 1,000 of them are aromatic plants, 350 species are rare species, and 2,400 plus are considered as a fodder plants in terms of agricultural field, and more than 350 are being used as a dye of yarns and etc.
Bu göstərilən rəqəmlər, o demək deyil ki, onları ədəbiyyatlardan sırf götürülür. Bu rəqəmlər Azərbaycanda xüsusilə mən öz tərəfimdən hazırlaşdırdığım və hazırda 20 PhD və 5 Doctor of Science elmlər doktoruyum. Dissertasiyaların yazılışında ortalığa çıxan rəqəmlərdir və bu məqalələrdən də görünür. Bunlar hər biri sayısı yüksək impact faktorlu məqalələrdir. Efir yağları, yem bitkiləri, dərman bitkiləri, etnobotanika və s. bitkilərin kimyəvi tərkibi, bioloji fəal maddələrin alınmasına aid 350-dən çox məqalə və kitablar nəşr olunmuşdur.
These numbers that I mentioned are not just cited from the literature. Under my supervision, 20 students have defended their PhD thesis and 5 students defended their Doctor of Science thesis. And in their dissertations, and due to their researches, these numbers came out, and more than 350 articles have been published based on those researchers, especially the aromatic essential oil plants, aromatic plants, and obtaining chemical substances from them.
Bundan əlavə patentlər alınmışdır. Dərman preparatlarının istehsalı üçün 17-ci ildə aldığımız immun bərpa edici patent sırf Azərbaycanda geniş yayılan bitkilərdən alınmışdır ki, və bu bitkilərlə COVID-19 pandemiyasında mən ailə üzvlərimi və yaxın adamlarımı immun sisteminin artırılmasında geniş istifadə elədim.
We also obtained patent about immune recovery property of some medicinal plants, which I have used in our family members. We have used them during the COVID-19 pandemic.
Azərbaycanın dərman bitkilərinin öyrənilməsində yalnız Azərbaycanın sərhədləri daxilində deyil, qonşu ölkələrlə bircə də tədqiqatlar aparılır. Bu gördüyünüz slaydda Təbriz, Naxçıvan və İqdirdə olan dərman bitkilərinin istifadələri, onların bir-birinə nə qədər yaxın olduqları deməli göstərilmişdir və qeyd edim ki, burada editor Münir Öztürk olsa da həm də bu əsərin həm müəllifidir.
The research on medicinal plants of Azerbaijan are not only going inside the borders of Azerbaijan, we also we have a thick cooperation and collaboration with the neighboring countries. As you can see in the slides, there was a research on Tabriz, Nakhchivan, and Igdir, the southwest eastern part of Turkey. And the editor of the publication was Münir Öztürk.
Və eyni zamanda İranlı həmkarımız Bəhməz Aslanpoyur və Təbrizdə olan bitkiləri Naxçıvanla müqayisə edilmişdir.
Also, our fellow colleague from the research, the doctor from Iran.
Və nəticədə eyni xəstəliklərdə bitkilərin istifadə tezliyi öyrənilmişdir ki, eyni bitki ilə hansı tezlikdə istifadə olunur Təbriz, Naxçıvan və İqdirdə və bu bitkilərin bəziləri hazırda İranda bir neçəsi artıq dərman kimi istehsal olunmuşdur və orada mənim də bir hissəm var.
We have
Xüsusilə şeker xəstəliyinin stabilize olunmasında.
we have comparatively analyzed the native medicinal plants from different parts, at which frequency they are being used by the traditional population. And after this research, some medicines are also being manufactured in Iran, especially for the curing of the diabetes.
Bundan əlavə xarici digər ölkələrlə 7 ölkə alimi birləşərək COVID-19'da deməli yeni bir preparatın hazırlanmasında mənim iştirakım olub. Həm məqalənin həm müəllifiyəm orada, həm də həmin məqalədə ondan çox istinad verilib mənim işlərimə və nəticədən məlum olub ki, Azərbaycanda olan Gliseriza glabra, Artemisia absinthium, Menta piperita və digər növlərdə qarışıq bir dərman preparatı almışdı ki, məhz bu preparat COVID-19'a təsir eləyən və o virusu məhv eləməyə qadir bir preparatdır.
During the COVID-19, in collaboration with joint collaboration with 7 countries, we have made up a new medicine which has consisted of medicinal plants, which is glabra, Artemisia absinthium, and Menta piperita, and it has an effect to killing the virus. We have published an article, more than 10 plus citations that article get.
Qarabağ haqqında bizim hörmətli səfir də müəyyən qədər məlumat verdi. Bizim Qarabağ bölgəmiz 30 il işğal altında qalmışdır bildiyiniz kimi.
As our Excellence Ambassador mentioned, our western part, Karabakh, was under occupation more than 30 years.
Və mən qərar verdim ki, bu məsələlər barədə, yəni Qarabağın florasına və Azərbaycanın müasir florasına və Qarabağda olan məsələlərə dair böyük bir əsərlər hazırlayaq. Bunun biri Bulaşıq İnger nəşrində, çap bir formasında, yəni 100 səhifəyə qədər olan bir məsələlərdir ki, bunlardan biri sırf Qarabağın etnik insanının bitkilərdən istifadəsi, dərman bitkilərindən istifadəsi və eləcə də bütün faydalı bitkilərdən istifadəsinə həsr olunmuşdur.
After that, we have decided to deep down the investigation of the Karabakh flora inside as a part of Azerbaijani flora. And after our research, we have published a chapter under the Springer publication. It was close to 100 pages. It includes what the traditional Karabakh villagers and the population, how they use the traditional medicinal plants.
Sual oluna bilər ki, Qarabağ işğal altındaydı, əhali qaçqın köçkün vəziyyətindəydi və bu məlumatları necə toplaya bildiniz? Bəli, 2009-cu ildən bir etnobotanika qrupu yaradaraq qaçqın və köçkünlərin məskunlaşdığı çadır şəhərlərində, yataqxanalarda onlarla söhbət apardım, xüsusilə yaşlı nəsildən və bu 71 kənd əhalisindən oldu və diaqramdan da görünür ki, onların yaş hədləri çoxdur orada. Yəni, yaşlı nəsil artıq Qarabağda olan bitkilərin istifadəsinə aid məlumatlıdır və onları gələcək nəsillərə çatdırmaq artıq bizim görəvimiz olmuşdur. Və buna görə də biz o məsələlərdən kitablar yazdıq. Qarabağ, Şərqi Zəngəzur və Qərbi Azərbaycan icmalarının yaddaşlarından xalqın unudulmuş reseptləri.
Maybe some would question if the Karabakh region was under occupation, how would you get all of this information about the traditional medicinal plants, how they were used? In 2009, we have set up an ethnobotany department, and as after this, after the occupation, so many people sought refuge from the other parts of Azerbaijan, and we have met them where they live, And in the population of 71 villages, we have conducted a survey, and as a diagram as shown in the diagram, the almost the old people know what the traditionally villagers used, what plants they used as a medicine, and after that we have published books about Karabakh and the Eastern Zangezur population about their traditional medicines.
Bu diaqramda həmin insanların məsuliyyəti göstərilir. Bəziləri xalq təbabəti ilə məşğul insanlardır, bəziləri fermerlərdir, kişilərdən, qadınlardan, təhsilli insanlardan və bunu böləndə aldığımız tam məlumatların 43 nəfər təhsilli olubsa, 80 nəfər insan isə oxumamış təhsilsiz insanlar xalq təbabəti üsullarından daha çox və daha dəqiq istifadə elədiyi ortaya çıxdı.
As shown in the diagram divided to the ethnic groups of the traditional peoples. There were traditional peoples, farmers, men and women, and 43 of them were educated people, and 83 of them were illiterate people. And also illiterate people have also deep knowledge about using of the medicinal plants.
Sözsüz ki, bunlar çox təbii surətdə aparılmadı. Müəyyən statistik analizlər və düsturlarla o rəqəmləri biz ortaya çıxartdıq.
We didn't do it as spontaneously. We strictly followed the methodology, statistical analysis, and some formulas in order to get those results.
Və nəticədə məlum oldu ki, hansı bitkilərdən Qarabağ ərazisində olan insanlar hansı tezlikdə daha çox istifadə ediblər və o bitkilərin burada faiz göstəriciləri əks olunmuşdur.
As a result, they have obtained the conclusion that which plants the traditional population of Karabakh was used at what frequency, and their percentile have shown in the slide.
Və eyni zamanda bitkinin bütöv orqanları, bəzi bitkilər tam yerüstü hissəsindən dərman kimi istifadə olunsa da, bəzilərindən toxumdan, bəzilərindən çiçəkdən, meyvədən, yarpaqdan, kökündən istifadə olunur və belə diqqət yetirsək, ən çox yarpaqdan və yerüstü hissədən tam istifadə olunduğu qərarına gəlindi. Yəni o statistik analizlərin nəticəsində.
As a statistical analysis, we get the conclusion that which part of the plants have been used as a medicine the most. We analyzed the aerial parts, seeds, fruits, leaves, and also roots. The most common used parts were aerial parts and leaves.
Və bu istifadəni də biz əsaslı eləmək üçün bitkilərin təbiətdə ehtiyatlarını öyrənməyə başladıq. Hardan toplamaq olarsa, əgər az yayılıbsa, onları toplamaqla məhv eləmək olar. Ona görə biz qərar verdik ki, biz bütün Azərbaycanda yayılan xalq təbabətində istifadə olunan insanlar tərəfindən tədarük edilən bitkilərin təbii ehtiyatların, eksploatasyon ehtiyatın və illik tədarük ehtiyatın öyrənəndən sonra əgər tədarükçü varsa, bitkinin yığımı ilə, satışı ilə məşğul olursa, biz onlara deməli, rəy verməyənə qədər Ekologiya Nazirliyindən onların yığımına qadağa qoyulur. Biz də o rəyi vermək üçün artıq bu bitkinin hardan və neçə ton yığmağını bilməliyik. Və bu nöqteyi nəzərdən bu istifadə üçün, xalq təbabəti də istifadə üçün bitkilərin ehtiyatları öyrənilmişdir.
And after this, based on those research, we have started to analyze the resources of those medicinal plants. We have investigated their exploitational and also yearly harvesting in tons, the kilograms and in tons. And because if it's a rare plant and it has a little amount of resource, it shouldn't be collected. And for now, in the Ministry of Ecology, if someone is about to collect and get himself or herself occupied with the collection and sale of the plants, they have to get a special permission from the Minister of Ecology, and Minister of Ecology directs that request to us, and after our permission, that happens. If we give the permission, which plant and how many kilogram in which part would be able to collect it or not.
Daha çok satışa çıkarılan bitkiler ise cədvəldə göründüyü kimi bitki orqanının hansı rayondan nə qədər yığmaq mümkündüyü öyrənilmişdir.
And we also investigated which part of the medicinal plants are in the most sale. As it's shown in the slide.
Bundan sonra həmin bitkilər laboratoriyada bitkilərin xam malından deməli müəyyən preparatlar hazırlayırıq. Düzdür, onlar sırf dərman preparatları deyil, çünki biz botaniki dərman preparatların Azərbaycan qanunvericiliyinə görə ancaq farmakologlar hazırlamalıdır. Biz qida əlavələri və yaxud da ki, losyonlar, demək olar ki, aromatik iyili maddələr istifadə edirik ki, bunları çayların ətirləndirilməsində, yeməklərə qatılanda insanların həzm sisteminin yaxşılaşdırılmasında köməkləsin və dezinfeksiya edici məhsullar da belə hazırlayırıq.
In our laboratory, from the raw materials, we manufacture the preparations, not the pharmaceutical products, because by law, it's only allowed for the pharmacists. But we, as a botanist, we make some preparations as food supplements, lotions, aromatics, and also tea additives, and some preparations for helping the digestive system, and also some disinfectant products.
Hansı bitkilər ki, təbiətdə nadirləşməyə, itməyə məruz qalır və o bitkiləri artıq biz əkin sahəsində əkirik. Botanika İnstitutunun 2 təcrübə sahəsi var. Biri dağlıq zonada, biri isə aran zonadadır. Burada aran zonada əkinlərin şəkillərini sizə nümayiş elətdiririk və gördüyünüz kimi pəncərə qarşısında toxumlar sertifikasiya olunur. Payızdan, yazda isə onların əkini ilə məşğul oluruq. Sonra insanlar bundan istifadə etsinlər ki, təbiətdə o nadir bitkilər itməyə məruz qalmasın.
We also investigated the rare plants that are available in nature, and also the plants under the danger of becoming extinct. We have 2 experimental areas. One of them in highlands and the another one in lowlands, as shown in the picture. From the fall season, the seedling process happens, and from spring season, the growth and harvesting.
Sözsüz ki, biz topladığımız bitkiləri herbariləşdiririk, herbari fonduna veririk. Botanika İnstitutunun çox mükəmməl bir herbari fondu vardır və Pakistanlı alimlərdən, Azam Universitetindən Məhəmməd Zəfər bu yaxınlarda bizim herbari fonduna baxış keçirdi və orada əyləşirdi. Amma onları sadə üsullarla quruduruq və ondan sonra o cür qablaşdırırıq. Çatan torbalarda, düzdür, biz onları hələ planlaşdırırıq, satışını da təşkil eləyək. Amma indi hədiyyə olaraq veririk. Hətta universitetdə sizin Azam Universitetində bizim bitkilərə aid Məhəmməd Zəfər professor sərgi də hazırlayıbdır.
After the collection of plants, we made a herbarium. Our herbarium foundation is one of the renowned one, and as one of the professor from Pakistan, Mohammad Zafar, also visited our herbarium foundation. We put those herbariums in linen packs. We also in the future we plan to sell them, but for now we present them as a gift.
Və Azərbaycan xalqını bitkiləri tanıtmaq üçün onları da aromaterapiyadan, fitoterapiyadan istifadə etmək üçün 24 kitab hazırlamışam ki, bunlar insanların hem rəhli foto şəkilləri var kitablarda, hem o biçilərin istifadə qaydaları, hansı orqandan istifadə olunur, neçə hissə qoymaq lazımdır və yaxud neçə qram. Bu barədə də geniş xalqı biz məlumatlandırırıq. Çünki biz rayonlara gedib biçi toplayanda, onlardan məlumat toplayanda çox qəribə belə rast gəlirik ki, bir bölgədə deyir ki, həm bu bitki ilə biz inəyin gözü ağrıyanda ora kompres qoyuruq. Digər tərəfdən soruşuruq, bu nədir? Deyir, alabotudur. Ona görə orada treyninqlər də keçirdirik ki, insanlar məlumatlansın ki, yaşadıkları coğrafiyada bitkilərdən necə istifadə etsinlər.
Out of the information that we have collected from the population, and in order to inform the people of Azerbaijan, we have published 24 books, and it was about aromatherapy and phytotherapy, the pictures and how to use and which part of the plant should be used. The receipts have been published in those books, and how many grams should you take, and the regions. It's one of the interesting fact that when we visit the regions in order to collect the plants, we also get the survey from the villagers and farmers. In one region, we got an information about a certain plant that they thought that they use it for the cure of the eye of the cow as a compress, but another region, the farmers told it's just a wild plant. So, and also we conducted some trainings of the different parts in order to inform the local people how to use the plants.
Bu ekspedisiyalardan fragmentlerdir. Birinci şəkil Savalan dağı İran ərazisində. Burada qayıq sürürəm. Görürsünüz, indi yaşlaşmışam, bəlkə sürə bilmərəm, amma vaxtında Azərbaycanın deməli 4900 metrə qədər hündürlüyünün 3668 metrə qədər qalxmışam yuxarıya.
As shown in the pictures, those were the expeditions. One of them is the Savalan mountain in Iran. As I get old age now, I'm not able to move up too much, but previously at 4,900 meters, I have elevated 3,668 meters.
Və bu fotolarda isə etnobotanika şubəsinin əməkdaşlarının bitki toxumlarının topladığı kadrlardır.
And in this photo, you show the colleagues of the ethnobotany department, they collect the seeds.
Təşəkkür edirəm diqqətinizə görə. Bu şəkil isə mənim doğulub böyüdüyüm Naxçıvanda Batabat yaylasının üzən adasıdır.
Thanks for your attention. And in this photo, you could see my homeland of Nakhchivan and the Batabat area.
Suallar varsa cevaplamağa hazırım.
I'm ready to get. I'm open for a Q&A session. Questions if okay, you give the question. What?
Əlbəttə var. Ənənəvi təbabətin özünün qanuna uyğunluqları var. Bunu Səhiyyə Nazirliyi tənzimləyir. Sözsüz ki, hər kəs bununla məşğul ola bilməz. Bunun üçün sertifikatlar verilir. Adətən həkimlər deyil, doktorlar deyil, loğmanlar bu işlə məşğul olurlar bilsin.
Yes, so of course we have a law about for been occupied with the herbal traditional medicines. Only the doctors, some doctors are allowed to use the herbal medicine.
Hətta onların toplanması üçün Ekologiya və Təbii Sərvətlər Nazirliyində xüsusi komissiya var. Amma sözsüz ki, rəyi botanika institutu əməkdaşları, mütəxəssislər verdilər. O bitkini toplamaq olar və hardan toplamaq olar, nə qədər toplamaq olar.
And in terms of the collection of the herbal medicinal plants, the one should have to get a special certificate from the Ecology and Ministry of Ecology of Azerbaijan, and that ministry is directing that issue to our botany and our specialists give the reference, give this permission.
Amma bu demək deyil ki, insanlar icazəsiz toplayıb satmırlar. Xüsusilə kənd icmaları bitkiləri yaxşı tanıyırlar və toplayırlar, satırlar, hətta bizə də gətirirlər. Qarşısını almaq biraz o qədər də asan deyil.
But it doesn't mean that no one are allowed to collect any medicinal plants. Some people, especially the villagers, still collecting the medicinal plants. They are selling it out of law, but it's not easy to regulate everything.
I agree with you.
Just at the lunchtime, we are already out.
Very short question. In Pakistan, second question is about clinical trials that Honorable mentioned. In Pakistan, I have requested Professor Dr. Shah, ICCBS Karachi University, Karachi, that the drug regulatory authority of Pakistan has no law to conduct clinical trials of muscle medicine. On my request, he given the job, and at that time I was chairman clinical study committee, and that has been approved, guidelines have been approved, and that's paid by the Shah. This is about, it's a legal document submitted by whatever Shah to the draft, and it is approved by the Drug Regulatory Authority of Pakistan. My question is, you have written a lot of books on the clinics about the chemical medicinal plants. You have conducted clinical trials on the medicine analytical in the market.
Yes, by law we have, as a law we have permission to conduct the clinical trials. In order to get the patent, yes, we conduct the clinical trials. As one patent I showed in the slides, it has been through the clinical trial, and after that it gets the Euro Asian patent.
Thank you very much, madam, for the very insightful presentation and for sharing your important research with us. We also have the same plans as you have in Azerbaijan, and I'm really thankful to you for the presentation and for the excellent lots of research work. Thank you. Now I will invite our next speaker, Professor Dr. M. Raza Shah, Director, International Center for Chemical and Biological Sciences, University of Karachi. We will be talking about supramolecular approach in drug delivery system and introduction of the ICCBS.
أعوذ بالله من الشيطان الرجيم. Let me thank the organizer who just provided the opportunity to share some information about our center and about the clinical ecosystems which we have at the ICCGS. Actually, initially I was supposed to talk about supramolecular approach and drug delivery system. That's the area where I have been working for the past 2 decades. But my teacher, Professor Iqbal Chaudhary, told me that it's better that you talk about the clinical trials, and that is more relevant to the themes of this particular workshop. Yeah, you know that Dr. Durashid, who is sitting here, he was the Director of Pharmacy Services. And we initiated this clinical trial ecosystem in Pakistan back in 2019, practically. But the rules in the rules and regulation conducting clinical trial in Pakistan, but there were no rules and regulation in Pakistan. And then I was conducted a meeting at our center chaired by CEO Director at that time, Dr. Aslam, and Professor Iqbal Chaudhary and Professor Atar Rahman and myself. We were present during that meeting, and we were asked that we should develop rules and regulation. And Dr. Sheikh Ansar was at that time the Director of Pharmacy Services, and we were given that responsibility. So then that room and handed over to Sheikh Ansar, and then he modified it according to the requirement in 2017. The room was approved, and then it was implemented, and that is still the rules which are used by the regulatory authority, that is known as Bio-Study Rule 2017. Then until the pandemic, we should conduct clinical trial. We are having the first drug approved clinical trial unit at the ICCGS. We initiated a clinical trial on herbal medicine. This is a traditional Chinese medicine. It's similar to the Unani medicine in many respects. And that was the first clinical trial at Yunhao 20 capsule, in which we randomized 220 subjects, and they were the first clinical trial on traditional Chinese medicines outside China. And this trial were approved by the Drug Regulatory Authority, and we conducted the clinical trial at that time. In our center. So the ICCGS is a complex of 17 research buildings. It's a huge research setup. And Center of Bioequivalence Studies and Clinical Research is one of the center where we are conducting the clinical trial. So I'm not talking about the whole ICCGS system, but only focusing on the clinical trial. Then we also conducted a clinical trial at Tsinghua Gingan granule, and that was actually we conducted during the pandemic. This is actually a kind of granule similar to the Dushanda, which we are giving to the patient in Pakistan for the treatment of upper respiratory tract disease. And that is the Chinese company, Jaxi Chang. They developed it for the H1N1 influenza virus in 2009, and it was marketed in China. So then the government of Pakistan and the Chinese government, the high official level has a meeting in the governor house in the same province, and the government agreed to just test these Chinese and Pakistani patients. At that time, we had 19 subjects in China that the pandemic was not at that time entered in Pakistan. So then I was given the responsibility that I should just carry out this trial on the Pakistani patient first, and then probably that would be a good medicine to treat the COVID-19 patient. And this way, we conducted this clinical trial that was again approved by the committee, which were chaired by Dr. Burashif. So that we randomized 300 subjects in this, and the trial were concluded. We published the results, and this trial the second best clinical trial conducted on the traditional Chinese medicines by the World Federation of Traditional Chinese Medicine, and I was invited to China to receive that award in 2022. Then we went ahead and then we tried to just carried out clinical trial on other traditional Chinese medicines. The first one were a few Kangen capsules that were, again, this was actually tested against pelvic inflammatory disease, and this was we randomized here again 200 female subjects which were suffering from the pelvic inflammatory diseases. And again, this was one of the star product, and the results were again very good. We compared that with allopathic medicines, and this was a double-blind study, and it turned out that the effectiveness of the acute Kangen capsule was equally good as compared to the allopathic medicines which are traditionally used in Pakistan, and that is doxycycline and metronidazole in Pakistan is mainly used, but this was actually as compared to that medicine. Then we actually, I also conducted a clinical trial of Sinopharm vaccine, and that was at the beginning of 2020. Actually, the China developed the vaccine, and there was a competition between the Sinopharm vaccine and the Pfizer vaccine. So then again, we asked that we should swiftly carry out the phase 1 clinical trial of the Sinopharm vaccine, so that the Sinopharm just take the leads and just combating this pandemic. So that was the phase 1 clinical trial we also conducted in our center on the Sinopharm vaccine. And then in return, then the Chinese government have given us a gift of 500,000 doses to our government in return of this clinical trial. Then we also conducted phase 1 clinical trial of RHN1 drug that is approved by the Drug Regulatory Authority of Pakistan, and at the same time it was also approved by the USFDA. So this was the only clinical trial in Pakistan which were totally conducted in in Pakistan and approved by the USFDA. There are many other trials which were conducted in Pakistan, but they were conducted in several other countries. So this was the first one where the trial were only conducted in Pakistan, and I led this clinical trial in Pakistan. So these are some of the results. This is actually what is the clinical research services which we provide in our center. So these are some of the exhausted list of the activities which we conduct, and these are the various steps which a clinical, if you would like to conduct a top quality clinical trial to be published at the international level, so you will have to follow all these steps which rightly start from the design of the protocol, and also statistical analysis and case report forms, CRFs. And these are all the essential documents needed by any regulatory body. You need to just get approval of all these documents in order to get approval So, like starting, and then the financial aspect. So these are the, each regulatory standard document. And once it is approved by the regulatory body, then one can conduct the study as per the protocol. So this is actually the clinical trial is a science. That documentation, it's 70% of your efforts and assets, and it is just consumed by the documentation process. So it's documents and documents. So every steps, each step is documented in the clinical trials, and after the completion of the clinical trial, then you submit this to the regulatory body. We also conduct live clinical trial, and that is many of the pharmaceutical company in Pakistan, they just approach us for the post-marketing of their products, and we just help them in the designing of their protocol and also the monitoring and the data collection of their products. So that's this is again regulated science. We conducted clinical trial with some patient for our clinical trial. So we have collaboration with a number of hospitals in nearly all cities of the Pakistan. We have conducted the clinical trial and bioequivalence studies for all the international players which are working in Pakistan. Here, so these are some of our valuable plants. So we conducted clinical trials and bioequivalence studies for this company. Our center is the only clinical trial in Pakistan which is accredited by ISO 17025, and it is still to it is the which is, and the facilities which we have, it is a dedicated and custom designed facilities as per the guidelines of the WHO and FDA. So whatever is the WHO for a clinical trial to be there, we have just incorporated that facility. So some of the facilities are just shown here, like we can accommodate 110 subjects at one time. There is a specified area for the volunteer, cafeteria area, recreational area. All the drugs are stored in -80. All the blood samples are stored in -80 freezers, and these are. Then for the kinetic studies, we do have very strong bioanalytical sections. We are having state-of-the-art LCM, SMS, triple quad systems, and we do have these many in our center. Similarly, a large number of HPLCs, and these are actually inspected and approved by the Drug Regulatory Authority of Pakistan and Pakistan National Accreditation Council. So that's just shows that the success of our center is just because of a research ecosystem which is there in our institute. So we have very strong engineering section. We have a strong analytical section. We are a very strong biological section. So all these sections companies together, and they are behind the success of the 17 research buildings where we are conducting research, and these ecosystems, their work is a unit and under the single management. So that's the reason that whenever we initiated any kind of research activity, we become successful in the sense that all the research unit are under the same management and they are working in the same direction. Similarly, the people like standby ambulances, fire tenders, and other activities. Some of the studies which we initiated in back 2013, you can see that the rooms were implemented by DRAP in 2018. But before the implementation of the rules and before the requirement, we were conducting the bioequivalence studies and the clinical trials. So these are some of the clinical trials list which we have conducted. You know that I mentioned the clinical trial is highly regulated, and it's the documentation and the quality management system is extremely important because the medicines which you have, the subject that have been consumed, it is not available for the testing, but whatever is available is the documentation and your quality management system, and that what the regulator normally inspect during their audit, technical audit. So from the beginning, I have emphasized in the center is to build a quality management system, which is essential for the approval and at the same time for the generation of the quality data. So we are, and as you can see that our center is accredited from the Pakistan National Accreditation Council, ISO 17025, then 9001, ISO 9001, ICH, GCP, RG guidelines, CFR, Part 11, and by study 2017. These are all the guidelines which were adopted and implemented in our quality management system. So our quality management system, which right from the beginning I implemented in the center, is that we are having a full layer of documentation process, and that should be implemented if you would like that your clinical trial is approved by any regulatory body. So you should develop a system based on these 4 layers of the documentation where we have the policy method. So these procedures should be documented. Construction should be documented, plans and records should be there. And these are actually the key level of the documentation which we, in order to just get approval of your clinical trial, you will have to implement these 4 layers of documentation. Otherwise, no regulatory body will approve your trial. No, that is what publishing a clinical trial is easy. But to get approved a clinical trial by a regulator, it's an extremely difficult task to convince a regulator, even in Pakistan. So that's what with this, let me thank you. I finished right in time. Thank you very much for your attention and listening me. And I'm sorry, I'll just share this topic, but I was just asked at the last minute that I should present the clinical trial unit of ICCVs. Thank you so much.
Thank you very much. We can take a few questions as we have 50 minutes. Yeah.
Well,
So you talk about the clinical trials for medicine, but in medicine you have used with traditional herbal medicines or medical complexes of a specific allopathic drugs.
These are herbal medicines, traditional Chinese medicines. So we just selected only those which contain only herbs, not any animal or metal products.
Okay, so you this only conduct the in vivo, in vitro, or in silico study also of these drugs?
You know that the clinical trial is only conducted in vitro. The in vitro studies were already conducted by the pharmaceutical companies which have launched it in China. So they had all the in vitro data, and also preclinical data like the animal data. So these are a list of the documents and list of the data which we need normally ask. We get the data preclinical data. That is the three kind of data you need in order to get approval. One is the safety data, and the other is the efficacy data. So the safety contain the toxicological studies, and then there are a list of the toxicological studies which should be there in before you are going to submit your dossier to drive for the approval.
Okay, so which type of animal you have used, like albino rats or rabbits or?
You know, like we need several kind of this is actually for the animals, the rats, but at the same time we also need primates. So if you are not conducting studies on the primates, then still that might be acceptable. So that is the preclinical.
Thank you.
Thank you very much, Dr. for the wonderful talk. And we are proud of ICCBS. It is a world-class, renowned institute in Pakistan. And every facility is available in under one roof. That is really special. Now I would like to invite the third speaker, Professor Dr. Gulzara Adayeva, Head of the Department of Botany, MSERA. She will be talking about current problems and challenges of study of useful plants of Azerbaijan flora. Welcome.
Good morning, everybody. Bismillahirrahmanirrahim. My talk is about fungi. Probably there are not many people who is involved with fungal research here. And I will try not challenge you with many Latin fungal names. Just give short, brief information what we are doing, what kind of studies conducted at Institute of Botany in my college. So my talk will cover information about some pathogenic fungi and also some beneficial species associated with mainly trees in Azerbaijan, because this is our planet, and if we pay attention, only one of the third is covered in green, and which shows that we actually don't have green zone. We actually have very little green zone in our planet, and there are estimated 73,300 tree species, of which 9,000 still yet to be discovered. And this is where my little country stays, and the map of the country. It also was shown today by Professor Sayare Badullaeva. I just would like to mention that Azerbaijan is a small but beautiful country, and it has borders with Russia, Georgia, Armenia, Iran, and it also nestles on the Caspian Sea. And if we pay attention to the map, one of the third of the country is covered by mountains, and we have forests also located mainly in mountainous areas. Greater Caucasus is in the north part, and the east part is the Minor Caucasus, and we have Hyrcanian forests in the border with Iran Islamic Republic. And if we talk about tree and shrub species, there are about 487 species occur in the country. They are mainly deciduous tree species belonging to the family Fagaza, like Quercus, Fagus, Castanea, and there are also Carcinus, Populus, Betulus, Ainus, and
as a genera are well distributed. Talking about evergreen trees, we have a species only of Uniferous, Taxus, and Pinus distributed in the country. As I said, my talk is about fungi, and we know that the fungi include about, let's say, 150 described species already. But according to David Housecourse, they actually, the number of species is estimated about 1.5 to 6 million species. And so this means that few millions have yet to be discovered in the earth. And fungi have a wide range of dispersal mechanisms. They also have a special ability to utilize different substrate, so they can grow in many substrates, and that's why most of them are cosmopolitan. They have cosmopolitan nature, and but we all know that fungi, they are short-lived organisms, but due to their adaptation to different situations, they form a different symbiotic also relationship, like lichens and or which they form with prokaryotes, bacteria, and also with trees like mycorrhizal fungi. And if you pay attention over the last decades, due to the globalization in the world and also free movement of goods around, new introduction of pests and pathogens is evident everywhere. So along with so we know that most of microscopic fungi, they could be saprotrophic, but there are also some of them, like belonging to the genera Aspergillus, Penicillium, and Trichoderma, have special metabolites, antibiotics that could be successfully used against STP pathogens. And along with micromycetes, macrofungi, they also have big, let's say, economic importance. As we all know, they have been used since ancient time in treatment in folk medicine, in different diseases in folk medicine. So briefly, I want to say what we are doing in my laboratory. So we do continue sampling of fungal specimens to reveal biodiversity, fungal diversity in Azerbaijan, and specimens kept at the mycological herbarium of the Institute of Botany. There are about more than 12,000, which represented by 3.5 thousand various species. So we collect and identify specimens by using traditional microscopy, but we also do some DNA barcoding of specimens, especially for pathogenic species and rare species, in order to confirm their taxonomic positions. But this is mainly done in the frame of bacterial projects, international projects. We do population structure analysis of some pathogenic fungi. And just a few years ago, we also started to make our first attempts to find out more about bioactive compounds of fungi. So here I'm representing results that have been done in my laboratory since 2000. So we study Ascomycetes, fungi or associated with trees and shrubs. About 193 species was found belonging to 20 orders, 59 families, and 100 genera. And if you pay to taxonomic spectrum of this species, so we can see that Pleospiralis, Botryospiralis, Helicoides, Leopardalis, and Xylarialis dominate in the number of genera and species. And this a checklist of this fungi has been published in Sidovia just recently. And we also looked for the host species of those fungi, and revealed that most of those includes just 49 plant species. And we paid attention to the organs that the fungi were collected, and about 100 species were collected from leaves and 39 from branches. But the other part were, had less fungal species. And I have to also mention that some fungi have been found in more than one plant species. And talking about fungi, special pathogenic fungi, I would like to stop on ones that were well distributed in Azerbaijan. The first one, this is powdery mildew fungi. Everybody knows them, and it's clear to identify them by symptoms. They just form white cover on green parts of the trees, like leaves, petals, sometimes in flowers and fruits. And the ones that well distributed in Azerbaijan is Erysipa alkaloides, which could be found on Quercus macrantera, and also another Erysipa species that belong to the same genus is the Erysipa quercicola. And but on chestnut, we have found recently Erysipa, another new Erysipa species, and it was described as Erysipa azerbaijanica, which is also a rare species. And another rare species is the Philakinia articularis, found on Fagus orientalis. And because of this, they were pathogenic, and there is also one new species. We wanted to identify clearly its taxonomic structure. So ITS phylogenetic ITS was amplified for both Erysipa alkaloides, Erysipa quercicola, and also Erysipa castellanifolia, in order to see that they are different species, and you can also see bootstrap values on the branch point. So this is actual phylogenetic analysis for ITF region and 28S RNA orthogon. And this was published in Microscience. And another well-distributed species in Azerbaijan is the Kurbanaktia parasita, which is the invasive species, and it now occupies all. It spread very well worldwide, actually originated in China, then moved in the beginning of the last century to United States, and in 1937, it was found first in Italy and quickly distributed in Europe. But the difference is in United States, the fungi was infecting Castanea dentata, which was most susceptible to disease. So that was the reason of fungus killed all chestnut trees there. But in Europe, it's another species, which is the Castanea sativa, and it is less susceptible. And the one good thing that they have found hypervirulent strains of fungus. Actually, it's a fungal strain infected with virus called CHV, Cryphanesia hypervirus, which was good to cure trees. And here we can see different types of cancers, the healing cancer and active cancer, and also medial growth down pictures. And in Azerbaijan, it was found in 2004. So then first what we have done, we collected 15 isolates and first identified them based on morphologies, and the ITS analysis. Actually, we did sequence, we sequenced them by using ITS1 and ITS4 primers. Then both ITS regions, including 5.8S gene, was amplified, and it was clear that isolates from Azerbaijan clearly matched to those from elsewhere of world, like Europe, Japan, China, and so on. And this was published as first report from Azerbaijan. And then we decided to look closely to the structure of populations. That's why we can see here districts that mainly chestnut grows. And we monitored all these Castanea sativa populations there. And it was revealed that 7 districts
of 8 that has Cryphonectria parasitica growing on chestnut population. So next step, we decided to look closer to the population structure, and we did VC type, vegetative type analysis. So here you can see two petri dish with different view. So this was, we collected about 257 isolates, 196 was involved to the studies. And these isolates were paired with the tester isolates in all possible combinations. And here you can see in the first one, the cultures merged. There is no lines between those, so we accepted those as vegetably compatible. And in another picture, we can see barrage or let's say didn't merge. So they have barrage between colonies, and it was accepted as vegetably incompatible. And based on these results, we identified 7 vegetative compatibility, different vegetative compatibility types, one of which included most of fungi, about 185, and namely AZ1, and another 14 isolates belong to other VC types. We also looked closely to mating types of the isolates, so by using mating type gene, and 90 of those did amplify MET1 and 83 MET2, and there was also 22 that amplified both MET1 and MET2 genes. And we also looked to all collected samples under the microscope, and only the 20%, like 53 specimens, had SI and vital spores. And another analysis of the population structure was conducted to reveal their genotypes, and observed multi-lox genotype and estimated multi-lox genotypes for each district evaluated. And in the end, we find that there are 6 different genotypes occur within the population, of which the first one, CK-AZA1, included about 192. I mean, just like in vegetative compatibility types, it was the dominant. The thing as vegetative compatibility types and also genotypes were absolutely different from those found, let's say, in other parts of the world, but it was very close to the ones that from Georgia. So we came up with assumptions that probably fungus moved to Azerbaijan from Georgia. So we did conclusions that parasitic population in Azerbaijan has a very low VC type diversity, which will favor biological control, and also the widespread occurrence of sexual and asexual structures suggests that both reproduction type exist within the parasitic population. This was published in Forest Pathology. And as I said, we checked all fungal isolates, and the no CHV1 was evident, and unfortunately, and this information also published in Forest Pathology. And then we decided to go for viral characterization. I'm not going to talk about viruses here, and just a quick note that we found immunovirus and reported it, and also RNA virus, new RNA virus belonging to antivirus group. And it was suggestions that maybe these new viruses block the effect of the CHVs. That's why we cannot find in the population this fungus. We also looked to the Castanea sativa trees infected with Castanea sativa infected with Cryphonectria parasitica. If they have some other fungi that could also affect drying enders of chestnut, 12 other species was found. Most of them belong to the Ascomycetes, 3 to Basidiomycetes, and 1 Mucoromycetes. So we did a conclusion that obviously Castanea sativa tree host more species co-occurring with Cryphonectria parasitica, but further research is needed to find out their effect to the tree. And just recently, we have started to study of bioactive compounds. I mean, actually, to find out if we have mushroom species that may have some bioactive compounds. So this was we first started with literature search, and we have found about 23 species growing in Azerbaijan. That this is a preliminary research. May have bioactive compounds that could be used for pharmaceutical or some other purposes. And this information based mainly on folk medicine. And here you can see that just a few species belonging to Markella and tubers, they are ophthalmitis, they have some immunoinflammatory, antimicrobial, anti-inflammatory abilities, and there are even more basidiomycetes that could be, may have different basic properties like proteins commandos. Okay, so I am finalized. I am not going to talk about the fungi separately because of time limit. And I just quickly would like to mention and thank the general coordinator of comstech, Professor Iqbal Chaudhary, and Dr. Atia Tulwahab for hosting my student Yabut Yusifova in Karachi University and helping her find out about bioactive compounds. I mean, to make first attempt on studying fungal bioactive compounds. And this research came out with good results. So a new secondary metabolite was found from crude extract of sulfureous, and also a new derivative was synthesized through biotransformation of anti-cancer drug dexamethasone, which also showed significant aromatase inhibition, but slightly less than drug dexamethasone. And so sulfureous is accepted as a source of bioactive secondary metabolite. So this just recently published in the journal Steroids. And I am finishing with this, just a conclusion about fungi that they shouldn't be, it's a wide group of eukaryotic fungi, and they affect different ecosystem processes. They are part of biodiversity, but at the same time, they affect highly to the ecosystem processes in different ways. But we shouldn't remember them just like the pests and pathogens, but we also should look closely to them in order to find the ways to benefit of them for human beings. And I would like to thank again comstech for holding to extend my heartfelt thanks to comstech for organizing this insightful workshop, and I hope that our collaboration will go further. I would like to thank Special Professor Iqbal Chaudhary and Dr. Afiat Yusufahab once more, and I would like also to extend my thanks to all my colleagues and collaborators from overseas and also from my country participating in the projects on fungi. Thank you very much.
Now we can have one question only. Yes. Yes, please.
Okay, I have a quick question. As you have mentioned that you have collected the same type of fungi from the different plants, right?
Can you please a little bit slow?
Okay, as you have mentioned in one of your slides that you have collected same type of fungi from the different type of plants. Okay, so what would be the possible reason behind either the same type of region or climate, or the plants share the same type of chemicals like the flavonoids or other chemicals? Okay, okay,
Let's question also, okay?
okay, as you mentioned that you have selected same type of fungi from the different plants, right?
Yes.
What would be the possible reason? Same type, okay.
So, yeah, like, diversity of fungal diversity of our country, for all biodiversity. Plant diversity, microbial diversity, so fungi is a fungus of microbial diversity. But actually they are. So it's a way to is some of them mainly small plants, because there are other of soil of water. So biological diversity now not on our hand and short application or short lifetime is but some from from like say the climate change, the current ability. And but for much by now how many buckets and can look those and can and the best benefit from. Okay, some of them are edible, they have property, but some of them have that's really. And there are also some compost. They are the always but something like that. So by now, what we had since our staff one, then we try our work.
Thank you. Thank you very much, Professor Desara. Okay.
You have mentioned that there are different fungi, same fungi from different plants.
Yes.
So what is the reason behind?
What's the
Spreads.
Okay.
Spread.
So, some of the protein, some of the protein fungi, not one kind of. Fine. So they don't really accept. They have different. Okay, so they just correlate and they just from air or from pollution or from different. But therapeutic kind of from plant. And there are some factors that for example, about the fact parasite on particles, but also some but both belong to the same so the segment was find the same. So but the parasite of the component. There are as about that just the comment has been all has of the
Thank you, Professor Dr. Desara. It was an excellent presentation. And thank you for the great contribution in the field of mycology. Thank you so much. Now I would like to invite Professor Dr. Atiyatul Wahab. No, I'm so sorry. It's Professor Dr. Nidar Mursal. She is working at the Scientific Institute of Botany, MSCRA. Her topic is research on fungi in Azerbaijan, emphasizing forest pathogens and mushrooms with biological activities and medicinal potential.
Thank you very much. My presentation is dedicated to the endemic species of Azerbaijan, their biological active compounds and the possible use. Firstly, I would like to introduce Azerbaijan's flora and endemic species. As you know, Azerbaijan diverse climate, soil, and vegetation, and its location is from the southern East Asia, from Asia, Iran, Iranian, and the Caucasus have looked at rich flora in Azerbaijan. It is enough to note that also the Republic of Azerbaijan accounts only 16% of Caucasus territory, but in Azerbaijan flora, you can see and found 30% of the higher plant species. This study of endemic plants is of great scientific importance in the formation of flora and in solving modern problems of the evolutionary process of vegetation. Many prominent botanists have contributed to the study of the endemic species in Azerbaijan, and as a result of many years of this investigation, there were 700 endemic plant species, but the latest information and the literature data in Azerbaijan flora have been determined 240 and 260 endemic species. But however, due to the recent opening of borders and the intensifications of tourist arrivals to the country, some species called endemic have been found in other countries, and they lost their endemic status. According to the work of Flora Azerbaijan, endemic species consist of 260 species, but to the latest literature, which is published in 216 years, the endemic species was reduced to 143 species. And the medicinal flora of Azerbaijan also included 1,600 species, belongs to 700 for genera. Among the medicinal flora of Azerbaijan, mostly representative of the Mangalepita species, and the leading families in this class, such as Aspirata, Fabasta, Lamiata, Rosacea, Apiata, and the Brassicata. And the leading genera of endemics of medicinal plants are Euphorbia, Salvia, Centaurea, Viola, Gallum, Stachys, Erythrina, and so on species. It was established that among the medicinal plants of Azerbaijan, the following species prevail, in which the following were found: flavonoids, mostly species consist of flavonoids, alkaloids, vitamin C, essential and effective oils, tannins, saponins, coumarins, and steroids. It also, we can say that among the medicinal plants of Azerbaijan, the species with curative, antibacterial, anti-inflammatory, and other properties prevail. Taxonomic species of medicinal plants you can see in this table, and there are totally 178 family consist of this family, and genera 740 and mostly 1,000 above 600 species. Leading families which include medicinal plants are Asterata, Fabasta, Lamiata, Rosacea, and Basta family. And also leading genera, you can see Euphorbia, Salvia, and the Centaurea. And distributions of medicinal plants mostly in Greater Caucasus, Kuba part, and most of the species are found there, and also mountain parts, and also Lesser Caucasus, North and Central parts. We also touched upon the issue of endemic and relations along the endemism of among the medicinal plants of Azerbaijan, and it has been established that of total number endemic species of medicinal species are endemic to Azerbaijan are 6 species, which is the endemic of Azerbaijan, and there are Anaphyllus silicatus, Cutularia kayagina, Penicillin gallica, Rosa azerbaijanica, Thymus karamarjanicus, and 77 species belongs to the Caucasus endemic. And most of the Caucasus endemics grow in the regions Greater Caucasus. Kuba part there are found 50 species. Greater Caucasus West part, and then Western Caucasus and Natural Mountain. The vertical zone starts from the lowland and rises to the high points in Caucasus, but our investigations range from between 20 meter above sea level to the 2,200 meter above sea level. This included lowland, foothill, low high mountain zone, middle and high mountain zone. In these pictures, you can see Eremus azerbaijanicus, which is the endemic plant of Azerbaijan. This is the genus Eremus included in the family Asphodelaceae, and has more than 60 species in Eurasia. But in Azerbaijan, this genus has two species, Eremus spectabilis and Eremus azerbaijanicus. Eremus azerbaijanicus also rare plant of Azerbaijan, and it was included in the third edition of Red Book of the Azerbaijan Republic, the third edition. And during field research conducted by us in Ismail region in May of this year, we studied of these plants populations in the village of Kur village in Ismail regions, and the useful properties of these plants and are in the treatment are used in the treatment of many diseases. They have pharmacological activity in vitro and in vivo. Modern pharmacological studies show that the various parts of species of the genus show significant antimicrobial, antibacterial, antioxidant activity. Young leaves of a number of species are in crooked form. Phytochemical studies show that these species contain flavonoids, terpenoids, coumarins, and especially in emergency tablet species is used in folk medicine for the treatment of jaundice, stomach irritations, diabetes, skin infections, and other diseases. This is Anacyclus ciliatus, which is belongs to the Astraceae family. It distributed in Bobstan, Aras lowland, and steppe palisades. And the content of biological active substance is essential oil, and it's used in traditional medicine and traditional medicine of medieval Azerbaijan, and used for treatment of some diseases such as toothache, paralysis, neuralgia, rheumatism, gout, and so on. And used part of this plant is are roots and seeds. Pyxilcan also the endemic plants of the Caucasus, and it's distributed in many parts of Azerbaijan, and it is used for the much more disease treatment, diarrhea, stomach, kidney stones, anemia, and etc. And a part uses that as bark, leaves, juice of fruits, seeds of this plant. Pinus eldarica, this is also native endemic plant of Azerbaijan, and it's distributed only in steppe plateau, Khanlar district, in Eller Orgu. And also listed in the Red Book of Azerbaijan, and content of biological active substance are vitamin C, collagen essential oil, and other components. Application area is the traditional medicine, and it has anti-inflammatory, disinfectant properties, and used part mostly needles and young fruits. Pituitaria caryagin also is distributed in one place in Nakhchivan mountain, and its biological active substance is essential oil, tannins, flavonoids, and use it only roots for the treatment some. Thymus caramarjanicus also distributed in steppe plateau in mountain, and in application area is experimentally studied, and they affect the properties of these plants, affect antibacterial and fungicidal activity, and used part also used is used mostly grass above ground parts of herbaceous plants. Atropa caucasica is the endemic plant of Caucasus, and You can see it is distributed in the most part of the Caucasus and also in Southern Lankaran and Western Caucasus, Greater Caucasus, and they have alkaloid content of biological active components such as alkaloids, thiamine, aquafine, and carotenoids, and so on. It is used for the more treatment of more disease such as gout, cold, poisoning, eye impotence, and also kidney liver. And used part of this plant also are grass, leaf, and roots. Senecio was also distributed in Greater Caucasus, and Senecio exhibits a range of pharmacological properties, including potential antibacterial, anti-inflammatory, and antioxidant activities, and mostly extract and isolated compounds from the Senecio species, including Senecio platypus, demonstrate the broad pharmacological effects such as antibacterial, anti-inflammatory, and antioxidant activity. I would like to thank all comstechs, mostly, firstly, to dear Professor Iqbal Chaudhary and his teams, Dr. Akira, Dr. Ajima, and other dear peoples, and other, and I would like to thank to other Pakistan colleges which have many good collaborations, such as Professor Mohammed Zafar, Professor Mushtaq Ahmed, and other people. I hope that our collaboration will be strengthened in the future. Thank you very much.
Can I have one or two questions? Okay, I have a comment for you. You have actually done an impressive amount of work. And if you digitize your plant in open rebellion, then it would be wonderful this in the United States. And like I have uploaded almost 600 plants of the letter plants. So then we can share that.
Yes, yes, I will do.
Thank you very much. It's a wonderful thing. Now I would like to invite Professor Dr. Atiatul Wahab from Dr. Panjwani Center for Molecular Medicine and Drug Research International Center for Chemical and Biological Sciences, University of Karachi. Her topic of research is chemical diversity and therapeutic potential of natural products.
Thank you so much, madam, for your kind introduction. So, we are here to discuss about the beauty of nature and the diversities that nature has given us. And this beautiful scarf that I'm wearing is also naturally dyed and gifted me by dear Professor Ciara. And it's a Azerbaijani scarf. So this is one of the use of our beautiful nature and natural compounds. Natural products, Newman and Craig, they are the very renowned natural product chemists. They have mentioned in one of their articles that natural products possess enormous structural and chemical diversity that cannot be matched by any synthetic library of a small molecules, and continue to inspire novel discoveries in chemistry, biology, and medicine. They are evolutionarily optimized drug-like molecules and remain the best sources of drug and drug use. So today I will be discussing about one aspect of the beauty of nature or natural compounds, that is its potential as to develop as a drug candidate. And as we all understand, there are two types of the metabolites that natural products usually possess that primary metabolites. These are different type of sugars, amino acids, nucleotides, etc. While because of their circumstances and their environment, these natural these plants or marine organisms have synthesized there are secondary metabolites, and these secondary metabolites are very fascinating, and so many stereochemistry in it, and they are beautiful, and they in structure, and they possess a variety of uh, biological activities. So secondary metabolites include lots of classes like different types of alkaloids, terpenoids, flavonoids, saponins, and diterpenoids. So there are a variety of classes. And important of phytochemicals, there are different types, they can be used as a medicinal and therapeutic application, and they have nutritional benefit. They have herbal, they are developed as a herbal formulation, as Dr. Iqbal also mentioned, that in his speech in the morning, that 100 years ago, when there were no synthetic drug available, so before that, so every one of us, the humanity was relying on natural products, and they were treating themselves and used in different type of for cosmetics and dyeing agent, for everything. There were these natural compounds that were that human beings were using, and they are used for the drug discovery and development, and definitely these secondary metabolites can be used, and they are using to protect the crops and agriculture from different type of infection, and also the treatment. And here you could see there are many Nobel Prizes were given to the natural product, to the drugs that are developed based on natural products. Like you can see, in 1939, the Nobel Prize was given to Sevetone and Muscone, these two molecules. In 1947, another Nobel Prize was given for the discovery of morphine, strychnine, cocaine, atropine. And in 1952, another Nobel Prize was given for the discovery of streptomycin. And in 1945, discovery of penicillin would led the Nobel Prize. And very recently, in 2015, the Nobel Prize was given on the discovery of artemisinin and ivermectin. So all these fascinating molecules that we could see with lots of stereochemistry and many biological activities, and they are remarkable drugs. So they are actually isolated from and discovered from the nature. And nature is always serving as a source of new pharmacophore, and that you could see that biodiversity, and also it serves as a living resources, and edible sources and everything. And we have surveyed actually for the use of medicinal plants for different type of diseases in the province of Sindh. And this project was done under the supervision of Professor Dr. Iqbal Chaudhary as part of the team. And we and our team has actually went more than 8,000 villages in the province of Sindh over the period of 6 years. And then we collected the data from people who are using these, from local herbalists, and also there are some tip or, doctors who are using these herbal medicines. From all these data we acquired from and then documented. And the reason behind it, because we would like to protect the resources because of the urbanization, and as we are fast moving, and all these information that we are actually getting are from thousands of years, from one generation to another generation. People are forgetting about this. When we have now headache, we just go and take some synthetic medicine. If we have cough, we take some antibiotics. So we are, and doctor, we just go to the allopathic doctors. We are just going far. So we would like to document this so that can be preserved for our next generation. And these are a view for few photographs that our team has surveyed during and in this team, we always have a doctor, medical doctor, and pharmacologist, and anthropologist, and a botanist. So we have a variety of people in the whole team to identify the disease and how people are using, and to identify the plant. So we actually collected the plants which are in that particular rural areas as well. And these traditional knowledge is actually slowly and gradually, we these traditional knowledge that we have documented that can be used to identify and extract the plant material and then take it further for different type of evaluation in bioassays and preclinical studies, clinical studies. So, and in this way, the traditional knowledge and science can come together. So these are one of our lab where we have extracted all the plant materials that we have collected during our survey and saved here. And this is an example of molecular bank that we have. So as Dr. Iqbal also mentioned, that ICCBS has been working in many fields, but is being renowned and famous in natural product chemistry for last 60 years. And there are many good natural product chemists that are world famous that we have, including Professor Iqbal Chaudhry and Professor Taur Rahman, Professor Vikar, and Professor Malik. And they are these are very renowned people in the field of natural product chemistry. And over the years, these molecules that have been isolated from natural resources in our institute are fully characterized using different spectroscopic method, and they are deposited and in this bank, which is called molecular bank, and not only isolated but synthesized and derivatized molecule also there. So now I will give an example of our work, and actually I'm part of a research group of Professor Iqbal Chaudhry. So I'm here presenting the work of all our group that has been done over the decades. And here you could see that a beautiful molecules from Buxus papilosa that traditionally is being used to cure malaria and skin diseases, and there are many other diseases. So these triterpene have been isolated and reported from our research group, and delphinium group is one of the poisonous plant, and here delphinium and aconitum both have been worked in our research group, and we have reported many nor-diterpenoid or diterpenoid alkaloids and their activities. And Catranthus roseus is a fascinating plant, and that actually shows indole type, and terpene indole skeleton that you could see here, and with many stereochemistry we have reported, and Alstonia, another plant, there are fascinating molecule have been reported. And again, there are a species of Dinospora that shows the triterpene molecules, and veratrum, which is again triterpene molecule, is being reported from Withenia somnifera. Withenolides are here that you could see, and from Resia species, here you could see different type of indole type of alkaloids are here. So here, an example of another and the work of our group, that is Katsura species, and there are triterpenoids are here that we have reported, and not only the new molecule, but you could see here they are being tested against various biological activities. For example, here from anti-inflammatory and also from for cancer treatment. And here some other molecules from Katsura and the structure. So these are the work actually of many PhD students that have been conducted over the period of time in our research groups. And here now there are flavonoids that have been reported from Coniza, and nortriphenyl chloride that has been reported from Portia, and they are checked against the breast cancer AU565, the breast cancer cell line. And there are flavonoids from Arus and triterpenoid and glycosides from Strophalanthus species. So all these molecules were isolated, and we characterized it and then checked against different type of bioassays. And here, Gravia asiatica, in Urdu we call it falsa. So these are the antioxidant molecules that we have reported from these species, and these and here in the small font, it's the antioxidant potential which is mentioned here. And these are 5-Salus minima, and there are 5 salins that have been reported and checked against the for anti-leishmanial activities. And here compounds from Barbarous species, and these compounds were checked for butyrylcholinesterase inhibitory activity and anti-inflammatory activity. So butyrylcholinesterase is an important enzyme for that are involved in Alzheimer's disease. So there are many compounds that we have checked against, and here actually compounds from lichens and Pharmocrema kupri species. These are isolated from a lichen, and they are also reported for anti-glycation activities. And then our group over the period of time have reported many novel compounds. So novel compounds are those whose that type of skeleton structure has never been reported in literature. So that is why it is called novel. For example, here in this molecule, the novelty is the 1819 bis norclerendone is reported here for the first time. And here there are some example of the novel compounds. And here the novelty is an alkaloid bearing N1-methyl and C4 methyl ester substituent. This actually bring it the novelty. So there are many novel compounds that have been reported from our group, and this plant was collected from Koita as honorable chair. Actually, she is long time collaborator. Before I joined as a student, the Institute of Dr. Iqbal Chaudhary, and she has been so kind always that she provide us the plant material in huge quantity. And she's been a long time collaborator of our research group and very kind person as well, and always facilitate us in many things. So these are novel alkaloid reported from Estonia. And so mostly the novel skeleton is being confirmed using X-ray, because these are the skeleton which has never been reported. So NMR always, we get the structure from NMR, but it's always good to have the crystal structure. So, and here there are the new novel carbon skeleton again from Roskia species. And so I'll take a little bit faster. So I just give here many examples. Okay, thank you, ma'am. So I quickly go through another, taking an example of the herbal supplement that we have prepared for the treatment of Parkinson's disease. We know that worldwide Parkinson is a huge problem, and estimated about 7 to 10 million people are suffering with this, and it has several motor and non-motor symptoms. And motor symptom includes the walking disability, posture disability, and depression, anxiety, disturbed sleep, etc. And there is no such kind of treatment yet available. It is the management of the disease which we have, and along with the Searle team, Searle company, we developed this, and we have obtained the Pakistani and US patent as well. There are 2 supplements, and you could see the fascinating molecules that are actually involved in the management. And the results, there's 2 supplements that we have developed, and the results we actually, I would like to show here the pathological group. So you could see the score. These are the scoring, which actually the higher the score 2 to score 8 is 8 is the highest score after 8 rats are died. So scoring is also in human also there is a scoring criteria of Parkinson. So we actually have the pathological group, and we let itself recover, because that Parkinson is not the rat or disease. So we have to induce by rotenone injection. So we need to see if it is self-recovery or not. And then we have the drug-treated group, which we use Cinemed, and we could see the result overall 30% improvement in different groups. And then these are the results of our herbal health supplement, and we could see 55% of overall improvement. And this is the herbal health supplement 2, which also shows 57% improvement. And there are different neurotransmitters involved in this disease. So we have checked not only this, we have isolated the brain of the animals and checked dopamine and 5-HT level, and we really find, you could see here the normal, and these are the pathological range. And after the treatment of our supplement 1 and 3, these are very close to the normal, and as compared to this pathological group, that shows it's really working. And we have filed the application for the clinical trial. So there, and these are different things that we have developed. In conclusion, we have demonstrated that it's possible to transform the folklore practice of the use of medicinal plant into clinically useful product through appropriate preclinical and clinical studies. Initiating from bioactive substrate and drugs, we can generate even more potent chemical space as drug-used molecules. These are my colleagues who are involved in this. And at the end, I would like to thank first Professor Iqbal Chaudhary. He is my teacher, my mentor, and all the work that has been presented here is under his kind supervision. though I'm a professor there, but every time he's, mashallah, very much involved. And whenever he visits, and even he's not visited and he's here, we always call and make him engage in different scientific engagement, and his guidance, and it's very visionary. He always guides us and give us the right direction. And we discussed a day-to-day basis, the science and everything. He's guiding us. He's our mentor. So whatever I achieve in my life so far is because of his guidance, his supervision, and also ignoring lots of my mistake. And he always, besides all my mistake, even I cannot be as patient as he is. And Madam knows, because Madam is much more closer to Dr. Iqbal Chaudhary, and he knows him very well, that he's very patient person, and he has very much patience. And second, I would like to thank Comstech organization who invited me in this workshop, and I'm so thankful to Professor Ciara. She is an inspiration personality. When I visited her, she has showed me whatever she has in a very short time, all the books and monographs, and hundreds year old books, and it's so much preserved. And I was really shocked that it's 500 years old that you were showing me. So she has showed me lots of things. It's a fascinating institute that he's running, and Professor Dr. Dilzara, she's like my sisters, and all my colleagues. Thank you so much.
So no time left for questions. Thank you very much, Dr. Athiya, for the very impressive presentation. And I would like to congratulate you and your team for conducting extensive research in this field. Thank you. Now I would like to invite Dr. Emil Shekurulu, Institute of Botany, MSERA. She will be talking about chemical composition and medicinal importance of Lectuca species of Azerbaijan. Welcome, madam.
Dear esteemed professors, scientists, and researchers, I'd like to greet every single one of you, and thanks the Comstech for hosting us right here, right now, such a kind of great opportunity. And as a brief introduction, I'd like to move on to the task that I have done. The Lectuca species of Azerbaijan, their chemical composition and some biological activities. In short, I would like to mention in healthcare, currently more than 8% of the world's population still prefers to use mainly medicines of the plant origin, and 25% of the prescription drugs are still derived directly or indirectly from the plants. And the global trade in medicinal plants and their products was valued at 60 billion United States dollar in 2010, and it's moved up to 5 trillion by 2050. It's an estimated number. Approximately 70,000 plant species are used in the treatment of different diseases, but only 15% of them are studied in terms of their pharmacological potential. A large proportion of the estimated 391,000 terrestrial plant species haven't been phytochemically studied, and their pharmacological potential hasn't yet been studied. Despite the richness and diverse flora of Azerbaijan, only a small number of amount, 5,000 plant species belonging to more than 200 families have been chemically studied. To the best of our knowledge, for the first time, we have studied the chemical composition of actually two species, Lactuca cereula and Lactuca tetragica. that grown in Azerbaijan. As you can see in the pictures, left side is Lactica cerealia and right side from your point of view is Lactica toparica. And after the analysis of the roots, we have derived the terpene derivatives and fatty acid composition. As you can see, the fatty acid, triterpenes, tricarboxylic acid esters showed differently. And also, these are the GCMS chromatogram of the crystals obtained from the acetonic extract of Lactica cerealia roots. And again, triterpenes, tricarboxylic acid esters, and fatty acids have been obtained, revealed. And also, we have conducted the novel MSD SPME technique, that's microsteam distillation and salt phase microextraction technology on three parts, the roots, leaves, and seeds of both Lactica cerealia and Lactica totarica. These are their chromatograms. And these are the molecules that have revealed by the analysis of those GCMS diagrams. 26 components have been obtained from the Lactica cerealia roots totally, and this is the same procedure have been done for the leaves of Lactica cerealia. And those are the components of the obtained from the Lactica cerealia leaves. 40 components have been obtained from the leaves, and the fatty acids from the Lactica cerealia leaves done by GCMS flame ionization detector combination. The main ones were palmitic acid, stearic acid, linoleic acid, and nonadecanoic acid, and again linolenic acid. These have been shown as an ester form because we had to derive them as an ester in order to reveal them, in order to detect them in the GCMS and flame ionization detectors. But their original forms in the plants are the acid form, fatty acid form. And lastly, the Lactica cerealia seeds, the fatty acids. These 11 fatty acids obtained from Lactica cerealia leaves, as you can see, mainly palmitic acid, stearic acid, oleic acid, linolenic acid, and etc. And the next plant is Lactica totarica. We have derived, we have also analyzed by microsteam distillation salt phase microextraction technique on Lactica totarica leaves, roots, and also seeds. These components have been derived, revealed from its leaves, mainly, again, these all are volatile components, hexanol, heptanol, limonene, and linalool, nonanol, and etc. Some of them are used in fragrance industry, some of them are used in pharmaceutical industry. And yeah, so 34 components totally obtained from its leaves. And with regards to the roots, totally 21 components obtained from its roots. Again, eucalyptol, as it's a well-known component, 1.8 semiol, pacimine, and nonanol, hexanol, some aldehydes and ketone, those that belongs to these groups mainly, and also e-anetol is also a well-known product. And with regards to its seeds, we have revealed 16 components. Again, this belongs to mainly aldehyde and ketone groups, hexanol, octanol, nonanol, decanol, and linalool, and etc. And we have made a comparison of the components that have been obtained from both species. And as you see the plus, that means these, for example, hexanol have been obtained all three parts of Lactica totarica and 2 parts of Lactica cerealia. And yes, totally 16 components have been obtained for the similar And also we have done some of the little simple microbiological activity tests with various microorganisms. We have conducted with the total extract. And we have tested these on only one Candida albicans fungi, which is well known. These microorganisms all are pathogenic for humans. And we have tested across extract, ethanolic, acetonic extracts. We used for ethanolic and acetonic extracts. We use dimethyl sulfoxide in order to get them dissolved. And extract with aqueous extract are dissolved in distilled water. Also, we have done their negative controls, positive controls, and comparative studies. And the ethanolic extract has shown more antibacterial activity in comparison with the other ones. And the next species is Lactica tatarica. The same experiment has been done on it with the same microorganisms, the strains. And these are the antibiotics and also antifungal preparations that use as a positive control. And additionally, we have done the next one, the Lactica saligna. We have done some of its extracts, the microbiological analysis, and the tetracycline, ceftriaxone, clotrimazole, ketoconazole, and fluconazole, meropenem are used as a positive controls, and again, dimethyl sulfoxide and distilled water are used as a negative control. Totally 6 strains. One of them was Candida albicans. The others were all are bacteria, the human pathogenic bacteria. Again, the crude ethanolic extracts have been shown the most effective on the bacteria, and also the less effective were the aqueous extracts. And this pattern has repeated all of these species, Lactica, Seriola, tatarica, and salica. The all crude ethanolic extracts have been shown the most active ones against microorganisms. And I would like to thank every single one of you, and from on top of the general coordinator to the persons who work behind the scenes. I would like to thank every single one of you for a great warm welcome and for hosting us. Thanks.
Thank you. I think she is not here. A lot of compounds, active compounds have been reported from Lactica. Thank you. Now I would like to invite Professor Dr. Hilal Imanvi. She'll be talking about prospects of the use of biologically active coumarin compounds of plant origin and their derivatives.
Assalamu alaikum, dear conference participants. Gratitude for you. I express my gratitude for your management of committee. My presentation will be about protect for your biological activity coumarin companies of plant origin and their therapeutics. Coumarin is belong of the Benzopyram family from the many medical plants. Natural chemicals demonstrated in the wide spectrum of the pharmacological activity, including anti-inflammatory, anti-algalant, anti-cancer, antibacterial, antimicrobial casein kinase inhibitor, antifungal, antiviral, Alzheimer's disease inhibitions, neuroprotective, and anti-cancerogenic, alexins, and antihypertensive. There are very few students on the validity of the commodities. Therefore, further investigations are necessary to study the availability of different commodities which already shows good biological activity in previous studies. On the evidence of the various pharmacological properties, discuss present and overall review of the duration of enable and the biological capacities of commodities with future considerations of the essential. Plant synthesis allows collection of natural products can lead to secondary metabolism. Secondary metabolism has important ecological functions, supporting plant protections against microbes and the herbivores and the practical parts in the attractions of the motor activity. Humans are secondary metabolites as the source of the dryness, aromatization, flowing agents, and the wide spectrum of other. Now let's briefly review to the presentation of the result of disease conducted. Resistance work in the obtained in the individual stage of the structures of the species of the Heracleum sosnovski, Angelica sahochana, and the Medicinal distributed in the Azerbaijan. The determinations of the pharmacological spectroscopy, infrared, and the chromatograph spectrometry method to their identification and to the study of the possibility of use in the medical practice. Only the small number of the 4,500 specialist plants distributed in the floor of Azerbaijan have been recently studied. Therefore, these biological activities to be studied in order to obtain them individually is gained rushes from the both scientific and applied point of view. Commodity derivatives from the plant basis biological activity substance are is a focus of the attention for most resistance to the broad spectrum of pharmacological activities. Thus, the most resistance for the analgesic, antibiotic, effective dilator of coronary vessels, anti-malignant, and photosensibility protector of the commodity derivatives. According to the literature data, commodity derivatives identified to the most that 1,626 plant species, general and 100 families in the world. Professor Severi Badalova, the flora of Azerbaijan, the Acacia family, which included 76 general, 187 species, and 1 subspecies in constantly in the focus of the attention of reasons engaging in the study of the biological activity substances of plant origin to the importance of the commodity derivatives in the medical practice. It is known that penicillin was activity against malignant tumors. Other commodity derivatives included the plastinacin constitutes as a mixture of bergactin, spandin, and xanthotaxin,
The eruption constituted of the mixture of the blood captain and the Santaxin, and consisted of the mixture of the isopropyl and the blood captain, Saralin, consisted of the mixture of the Saralin and the Angelicin, and the Meladamin consisted of the Santaxin and the Infiltrarin, which are used in the treatment of the vitiligo. For the first time, three species belong to the family Apiaceae, widespread in the flora of Azerbaijan at the modern level, Heraclium sosnovski mandin, Angelica sapochena female, Amnigisnada, were studied using chemical chromatography and the spectroscopy methods. Six substances combined derivatives were individually isolated from the root of the Heraclium Sosnovski species, and the four combined derivatives were individually isolated from the roots of the Angelica sapochena species by column chromatography. The composition of the root Heraclium species, the roots and the aerial part of the Angelica sapochena species, the roots, aerial part and the flowers, together with flower umbels of the Angelica amnigisnada, plants in the Azerbaijan flora were studied using the gas chromatography mass spectroscopy method. 8 coumarin derivatives were detected in the roots of the Heraclium sosnovski plant, 15 coumarin derivatives in the fruits, 12 coumarin derivatives in the roots of the Angelica sapochena plant, 7 coumarin derivatives in the iron part, and the 21 substances from the various groups of the natural compounds were detected and identified in the flowers of the Amnigisnada plant. 89 components in the fruits, roots, and the 41 components in the aerial parts. This is Heraclium sosnovski mandin pictures, Angelica sapochena, Amnigisnada. As the objective study was Heraclium sosnovski, Angelica sapochena, and Amnigisnada, isolated of the some of the extinctive substances from projective of the disease was performed by the extraction method. Absence of the biological activity substance in the individual state from some of the extensive substances by chromatography method using the Soviet neutral aluminum flow. Oxygen 3 with activity 3-4, and emission using hexane benzol chloroform and the mixture of the various professionals. Verification of the individual of the substances with conducted over the method of the same layer chromatography on C4 with 255 letters. Identification knowledge substances was conducted using infrared NMR spectral method. The natural also functions of the characters of the substance was determined using classic chemical methods. The component composition of the various plants, Angelica sapochena, Heraclium sosnovski, and Amnigisnada, were determined using by gas chromatography spectral method. For this purpose and using an excellent technology, GSM system, chromatograph with the mass spectrometry, mass selective detector, and the Quadris capillary form HP 5MS methyl silicon. Individual coumarin provides Heraclium sosnovski mandin Sarayın, Angelis'in, Santa Tex'in bir baktığın, implementin, üzerinden, we see that infrared spectrum of 6 components. Sarayın, Angelis'in, Santa Tex'in bir baktığın, implementin, Kumarin derivates obtained in the from the root Angelica subordinate species. 4 substances. The one substances infrared spectrum, carbonyl offset beta electron cycle, NMR spectrum, Vegaptin, substance 2, infrared spectrum, and the NMR spectrum, xanthataxin, substance 3, infrared spectrum, and the NMR, pimpinidine, substance 4, oscillotol, infrared spectrum, NMR spectrum. GSM mass spectral of the characteristic components of solutes of uranium substance in methanol chloroform extract. This is the name of component angilicine, mass spectral angilicine, mass spectrum cerulean, mass spectrum pedonum, mass spectrum bergaptin, xanthataxin, pimpinidine, orgestolinum, is other 20 component. GSM chromatographic biochromatographic component is helicum substance to fruits, methanol, chloroform extract. Combine context of fruits and fruits of helicum substance, this combines the part roots and fruits relation time among components. There are components 14. They similar color. These components similar. Color is a different component. Because characteristical component is Heraclium Sosnovsky, pimpinidine, reaching point 20, very high, very captain, 26.3 to 5, angilicine, isopimpinidine, xanthataxin. Similarly, of the fruits, angilicine, 14.9, Sarayun 0.97, xanthataxin 4.48, Belgaptan 14.72, pimpinidine 15.54, and the isopimpinidine 7.40. GSM chromatogram of the characteristical component of angelical subproduction rules methanol extracted. This chromatogram combining continuous roots and the aerial parts angelical subproduction. Roots and the aerial parts. Mutation time among calcium percent. We see similar components. Color similar. GSM مثلا قرص methanol chrome extract was to medicinal part. combating context of roots and areas of the Amibisinada. The character of component of the Amibisinada is the high persons, bismagine, Belgaptine, Tekucin. This is Hamasamas Amibisnada of progress and the continued in this chromatogram, Meshamagram extra of the flowers Amibisnada. Commoding continuous of flower included Amibisnada. This is Kumarinus Angelism or Visnagin, Noragin, the characteristic component of Visnagin, Norogin, Chivlin, Norvisnagin. Composition of the characteristic components determined in the roots, flowers, and areas of Amibisnada. The single color is it three parts of the plant. Visnagin, Norvisnagin, Norogin, it is similar, and the estaphytin, chelone, linalol, cisneradol, longibernol, and farconol, and the pyrethrin different components. So chemistry is the study of bioactive plants focused on the chemical preparations and the biological activity. It plays a crucial role in discovering new therapeutic agents as many pharmaceutical arguments from the plant sources and analytical clinical such as chromatography, mass spectrometry, nuclear magnetic resonance spectroscopy, and infrared spectroscopy are essential for identification, quantitative and characteristical plant metabolism. The technicals enable regions is a way for components determines the molecular structures and assesses the potential for drug development. Pharmacological factors on the study of the natural particle from the plants as the third component. Quality control and standardization are essential to ensure the effective supply and the consensus of the herbal medicines. Methods included identity isolations, microscopic examination, and chemical profiling using technical such as the high performance chromatography and mass spectroscopy. Standardization involved
Excuse me, Professor,
determination.
can you please wind it up? Wind up. Just take one.
Just a moment. As the result of the conditions to that individual components from modifiers can be used in the preparation of new effective drugs in medicine. The activities of the pharmacokinetics components obtained from the plant species, which are considered important to practical medicine, are traditionally used socially in the treatments of the diseases such as anti-inflammatory, diuretic, stimulant, and diuretic against cold, influenza, hepatitis, antivirus, integrations, chronic bronchitis, typic fever, disease, chickenpox, MERS, bacterial and fungal infections, and the urinary tract Single from commodities have antioxidant, anti-tumor, photosensitization activity. They can be applied in the medical practice for the appropriate workplace. Thank you for your attention.
Thank you, Professor, for the outstanding contributions. Lots of work has been done. And that concludes the session. I'm thankful to all the speakers, honorable guests from Azerbaijan, and we look forward for further collaborations, particularly with me, because I am the botanist. But many others are chemists. So we can have lunch and prayer break now. Thank you very much for joining us here. Thank you all.
Okay, so we have a lunch break and it's the prayer time. So we join by 2:15 o'clock at the auditorium. Thank you. Okay, so it is our second technical session. I invite Dr. Mohammed Zafar for chair the session. He is the assistant professor at Azerbaijan University Department Plant Sciences. So please, sir, join the session.
Bismillahirrahmanirrahim. I would like to formally open the session. I would like to invite Professor Dr. Ashana Dar. He is professor from Hamdard University. The topic of her talk is need of evidence-based studies supporting traditional system of medicines, a challenging experience. So please, Professor Ashana Dar.
Bismillahirrahmanirrahim. Assalamu alaikum. Subhanallah wa bihamdihi, subhanallah wa. My name is Ahsana Dar Farooq, and I am a natural product pharmacologist. I served ICCBS, so Professor Iqbal Chaudhry has been my younger colleague. I served there for almost 3 decades as a natural product pharmacologist. After my retirement, I thought of working on traditional system of medicine, providing scientific justification of traditionally used plant. That's how I got into it. Although I was working in the areas of anti-inflammatory, anti-cancer, and antidepressants, using a variety of in vivo and in vitro systems. Today, I'm going to talk about the traditional system of medicine, which is practiced in Pakistan. In Pakistan, we have a system of medicine, traditional, which is referred to as Unani system of medicine. This Unani system of medicine is actually provide holistic approach to the understanding of the human body and the medicaments. And the father of the Unani medicine is Hippocrates. Later on, he actually got his students, Galen and the others to follow, who not only enriched the Unani system into a complete systematic form that flourished into a golden era and developed during the Arab civilization. Now, during the 13th and 17th century, Arabs not only saved much of the Greek literature and also enriched their own contribution. As you can see, it originated from Iraq, and, which was the seat of learning at that time, although it was originated in Iona, and it's a Greco-Arabic system. From there, it spread to the various parts of the world. Wherever it went, it was enriched by the traditional system of medicine, which was being practiced in that particular area. In Pakistan, as you are, so I welcome you here, and I hope my guests from Azerbaijan are enjoying their stay here. It's a beautiful country, and nature has blessed us with tremendous treasures, right from the north to the south, the Arabian Sea and the rivers. At the moment, we are suffering floods, which is very sad, but nevertheless, we'll come to control it. So we have great potential, and with that potential comes the responsibility. Coming back to the Unani system of medicine, in Pakistan, about 80% of the people, they rely upon this system of medicine because particularly in our villages area. Now, the main person who played an important role in flourishing, in establishing the United system of medicine education and practice is Hakim Said. And he actually, during the partition, he opted to join Pakistan. He came to Pakistan and established the 100 clinics, 100 laboratories, and of course, research centers, and later on, the university, which I'll talk to you later. Then the lady on my right is Madam Sadia Rashid, who is the chancellor of Hamdard University and is the daughter of Hakim Said, who is actually not only carried on the mission of Hakim Said by doing the philanthropic work and the educational work, and there has been a continuity, and she has also helped in the development of the Faculty of Eastern Medicine, particularly in the development of Unani laboratories, that is Unani system of laboratory system. And she is serving here. Gentleman on my left is Hakim Abdul Hanan. He is a very renowned Unani medicine practitioner and was the ex-vice chancellor. And the gentleman on the right is the present vice chancellor. He's doing his administrative job very efficiently. Now, the journey of the Unani medicine was initiated, as said earlier, by Hakim Said as a school, which was referred to as a college, where the education was free. Later on, then in 1991, the Hamdard University was established, and the Faculty of Eastern Medicine dealing with the Unani medicine was established in 1995. In Pakistan, we have the legal status of alternative medicine, and there was in the Parliamentary Act, which was then Unani, Ayurvedic, and Homeopathic Act in 1965. And there are specific councils for the homeopathic and for the Unani and Ayurvedic. Now, the Bachelor of Eastern System and Surgery, which is now referred to as system because the surgery has been replaced by the word system, is a 5-year program, degree program, with 1 year of clinical training. So you can see we have got a very impressive program, which is a bachelor's, which provides the bachelor degree in Eastern Medicine, and it has been approved by the various regulatory bodies. And of course, it's under the council, that is the National Council of Fed. And in addition to the undergrad system, we have got the postgraduate system leading to MPhil and PhD. So that means there is a lot of experience that the Hamdard University enjoys. And if anybody is interested, in particularly our guests from Azerbaijan, if they're interested in developing the curriculum and developing and initiating the program, the undergrad program and research collaboration. We will be honored to help you. Now we have in Pakistan more than 14 colleges, more than 14 universities which are offering this program both in the private and the public sector. As I said earlier, there are regulatory bodies, and most recently, on 26 February, National Council of PEB visited us and evaluated. Now, in this whole system, when I joined, they are excellent practitioners. Their knowledge is perfect as far as diagnosis and the treatment is concerned, and it follows the Unani principles. One thing which was lacking was the evidence-based studies as far as the animals are concerned. So keeping that in mind, I requested our chancellor that I want to establish a Unani medicine research laboratory. And that's how it all started. And then there was COVID, so we had to wait. Nevertheless, finally, the keys of that empty laboratory was handed over to us in 2021. The Unani system has got a certain structure, as you can see, and the person who is, I have highlighted with red mark, played an instrumental role in developing the animal models, particularly the ones which I've been talking about mainly. Now, he has his experience from China, and he has very strong background of physiology and immunology, and that is a perfect combination for the animal models. Now, this is the training of the UMR teams, because you remember I said that they have got Unani medicine background, but they don't, of course, they are practitioners, and you don't expect them to know the various intricacies which are involved with the animal handling and the pipetting and all that. So we have to train a group of people. Now, the research activities at the moment are varies from antioxidant, hepatoprotective, anti-inflammatory, and PCO. I will be talking about the PCOs. And the Unani formulation, which is referred to as Picolin by one of the graduate students who received her PhD just this year. Her name is Tabiba Lina Hamid. And she is, and the gentleman who is on the left side is the research supervisor, the one with the glasses. And this is the work I'm going to talk about. PCO is the most common endocrine condition in women of reproductive age. It increases the risk of the metabolic disorders, and about 80% of the women who are suffering from infertility, they have PCOS. Not only that, 60% of the women with hirsutism, that is the hair, also have also the possibility of developing PCO. The three major diagnostic criteria are the menstrual irregularities and the polycystic ovaries, that is, there are cysts there, and the clinical or biochemical hyperandrogenism. You see, the androgens, the level of androgens are higher. And there are 30% of these sufferers, they develop acne. And it's a very complex hormonal disorder with diverse symptoms. And any of out of three signs which has been shown, if they signs of symptoms, they might develop PCO. The global incidence varies from 4 to 20% in various areas. However, the reports in Pakistan is quite alarming. It's over 50%, and I think it's about 54%. Nevertheless, Tabiba Lina, she did a small survey of the medical students in the university, and she actually collected the data of 286. It's a clinical data which she collected, and there is the different parameters which you take into account for the clinical data. And 21% of the females, they suffered from PCO. That's very alarming. Again, The pathophysiology could be, is more dependent on the genetic susceptibility as it runs in the family. You can't do anything about it. If it is there, you are born with it and that's it, but you have to take the precautionary measures. The other thing is the lifestyle. Nevertheless, whatever the case may be, in the pathophysiology indicates high levels of the insulin resistance, visceral fat, and serum luteinizing hormones. Now, the reason is that the conversion of the testosterone to estrogen goes down. As a consequence, the androgen levels increases. It is a state of the hyperandrogenism, and androgen causes an aovulation, meaning that they cannot ovulate, leading to PCOS. Now, if you look at the Unani concept of the PCO, according to Ibn Sina, the characteristics of hirsutum due to an increase in the androgens, evolution, and amenorrhea, which is the same thing. However, the hormonal disturbances is because of the Balhami massage, because in Unani medicine, it's the massage which is very important, and that is phlegmatic, meaning that the nature of the disease is cold and moist. When it is cold and moist, depending upon the situation, the treatment would be just the opposite to that. So that treatment would be hot and dry. This you have to remember, because I will be talking a lot about it. Now, what was the rationale of this study? The rationale was due to the complexity of the pathophysiology of PCO, approach is symptom-oriented. Once it is symptom-oriented, that means one has to explore to the other systems in which there is a holistic approach, and Unani system of medicine does provide a holistic approach. For the management of PCOS, the picoline was formulated by combining various plant extracts, rather various plants that are traditionally used according to the temperament, that is hot and dry of the disease, and supported by the scientific evidence of decreasing the reproductive and metabolic disturbances which are linked with the PCOS. So keeping that in mind, she made this picoline preparation. Before that, our previous studies demonstrated that picoline has the potential to improve the hormonal balance in the PCO female patients. Remember that all the traditional system of medicine, including the Unani system of medicine, is time approved. It is time approved and it is human tested. Human tested, the evidence is there. The clinical studies are there. Time tested, nobody during even that time reported any ill effects or the side effects. So it was considered to be the same. Now, ingredients of picoline is, as I said, the massage being hot to dry too. The plants which are used are written in the ingredients and the part of the plant and the temperament of each plant and the evidences which are there for their pharmacological actions. And the voucher number is there and the combination of 5 kg. The aqueous extract and the capsule preparation of the picoline for the animal studies, ingredients, it was a extract that was soaked in the hot water and filtered. However, for the clinical studies, because people don't prefer to drink extract, so it was put into a capsule. The first thing to do was to look at the safety profile, because the main thing is safety, efficacy, and standardization. So when the safety was done, using control animals and the picoline treated animals, and what was monitored is the liver function which was monitored, and the kidney function was also monitored. That is the enzymatic levels. And there was no difference between the control and the treated group. So it was safe. Once it was decided that it was safe, then it comes the efficacy of the picoline. And the animal model was then prepared in which the androgens and using the letrozole, which blocks the conversion of the androgens to the estrogen. Okay. And this PCO model, which was prepared by using a third generation aromatase inhibitor. And one of the speakers talked about aromatase inhibitor in her presentation. I remember she used, when she was looking at the enzymes, aromatase was also there. Now, this is the first evidence based on the Unani system of medicine in animal-induced PCO model in Pakistan. And it is the first time that the animal model was developed at the Faculty of Eastern Medicine from that university, and this was done under the guidance of Professor Numan Khan. So the groups are such that group 1 was untreated, that is a control group, distilled water. Group 2 to 7 was PCO-induced group in which the letrozole was administered 1 milligram per kg daily for 28 days. And this is the duration of the experiment from day 1 to day 28. And then all the levels of the written here were then measured. And once the PCOs developed confirmation was done, then the treatment was treatment was started. So once the treatment was started, so I have to hurry up, I have got just 2 minutes. I'm very surprised. So the different stages of the estrogen cycle was then noted, and these are the different stages of the cells, that is the prosterus, esterus, and all that, which is written here. It is, in the case of the PCOs, it was the esterus cycle, which was then, actually, we can see here the effect of picoline. Control was there. The cycle is repeated after every 4 days. In the PCO, you can see it was blocked in the distress. The cells, these are the cells which are collected from the vaginal smears and then monitored. So in the stress cycle of the PCO, it was consistent pattern was delayed. You can see the line, the line which is there. Here, this is the plateau. You can see that that it was picoline has reversed the condition, whereas in the case of the PCO, this is the model, it was reversed. It stayed with, it was reduced here. It was also reduced by the metformin, which is a standard drug which is used in the PCO. Now, in the case of this diagram, where you can see that the hormonal levels, the black one shows the model. So in the model, you can see all the levels are raised. Whereas in the case of the picoline and in the case of the metformin, it was then reduced. Effect of picoline and the serum glycerides, you can see that again in the case of the black, which is the PCO model, the levels were raised and which were then reduced in the presence of the picoline and the standard drug. You can see in the PCO levels, the ovary weight, the PCO model, the ovary weight, the number of follicles, and the number of cystic follicles, and you can see they were raised as compared to the control system. I'm sorry to say the number of participating was reduced. However, in the case of the metformin and picoline seems to be more effective. Now, these are the pictures which again shows the differences between the control on the PCO model and the treatment. And we can see that the levels are there. Conclusion being that it is non-toxic. It is actually, picoline seems to be more effective than in normalizing the hormone.
levels as compared to metformin. Histological changes in the ovaries are associated, and these findings suggest the potential therapeutic benefit of the picoline. Now she has received many awards and she has been very active in generating awareness. Now we are going to talk about some of the antidepressants. Not since Nina developed this background of handling the animals, being a tabiba herself. She then started her own group who are doing now research in the field of the antidepressants, forming the in vivo studies and behavioral studies, biochemical studies. Then another girl is Aisha. She's doing the antimicrobial activities, and they are now generating evidence-based studies using microorganisms or the animals, and the potential of the extracts for the prevention of lung fibrosis is being done by another student. So they are using animal models to induce the lung fibrosis by the bleomycin. Bleomycin actually is anti-cancer drug, but it also induces the fibrosis in the lungs. And then she is also working with the grapes and grape leaves are providing evidences that it has got the antioxidant potential and is also going to antagonize the respiratory ailments. Then there is a hepatic protective studies, which is also being conducted by another MPhil student, and they are all combining their work together using a combination of in vitro and animal studies. And this is the evidences of their studies. And long live Pakistan, Azerbaijan friendship. And I do hope you recognize the beautiful hairy bulbul and yasmin. These are the national flowers of Pakistan and Azerbaijan. In the end, I would also like to thank all the audience for their patience, for putting up with me. And I would also like to thank Professor Iqbal Chaudhry for inviting me here and for giving me the chance to talk about this system. But I'm not a talker for 20 minutes. I like to talk for more than 1 hour. So that is my problem. So I want to explain things and try to make everybody understand. Nevertheless, thank you very much.
Thank you so much, Professor Sanadar, for your briefing about Hamdard University, history of Hamdard University, also about Hakim Muhammad Said Saab, also difference between Unani Ayurvedic and homeopathic council. Also, you are pioneer in UMRL. So just very brief questions, 1 or 2 questions from Professor Sanadar. Anybody, please?
Well, if you don't have any questions, I have.
Okay. Yes, please. Please, Doctor.
I would invite both you come and visit Hamdard University and have a look. We have got 7 faculty where you will be entering. And would like to invite in particular. And I would like. And I would like to invite in particular our guests from Azerbaijan. By the way, I visited Azerbaijan and I have been to Baku and one of the conferences. It's such a beautiful place. Thank you very much.
Thank you so much. So our next speaker is Professor Dr. Jamshed Iqbal. He is a renowned pharmacologist. She is representing Pharmaceutical Sciences, Comstek University, Islamabad, and Ahmedabad campus. So the topic of his talk is discovery of new ligands for ectonucleotidases to explore their potential for treating diseases. It's a very interesting topic. So please, Doctor.
Bismillahirrahmanirrahim. So welcome all of you and have a good afternoon. Our topic is discovery of new ligands for purinergic receptors and ectonucleotidases. to explore their potential for treating different diseases. So this was the review published in the Nature Drug Discovery. It shows there are two major targets. One is enzymes and the second is GPCR receptors. So 28% drugs, they target the enzymes, and the major is the receptors. So our group is mainly focused on the receptors and the enzymes, but they are more closely related to each other. So we are mainly focusing on the purinergic signaling. So we have the cells, when it is injured or in the different condition, it releases the adenosine triphosphate outside the cell, and then it is phosphorylated, dephosphorylated by the different enzymes, ATPases, and then ATP is the agonist of the receptors like P2X receptors, P2X1 to P2X7 receptors. Then it is dephosphorylated to adenosine diphosphate, and it is again agonist of the P2Y receptors. So then it is again dephosphorylated another class of ATPases enzymes, nucleotide diphosphate diphosphohydrolases, to 5-AMP, adenosine 5-monophosphate, which is not agonist of the receptors. However, when it is dephosphorylated with that to 5-nucleotidase, it generates the adenosine, which acts as the agonist of the P2-1 receptor. So we have different receptors and different enzymes. They are located at the cell surface. So it is our goal to develop the selective agonist or the antagonist of the receptors, as well as the inhibitor of the enzymes. So in the purinergic receptors, there are three main classes: P1 receptors, which is adenosine A1, A2A, A2B, A3 receptor, and the P2 receptors, P2X, which are the ion channel receptors, and P2Y, these are the GPCR, glycoprotein coupled receptors. So we don't work on the P1 receptors because they require the radioligand binding assay, so we don't have the facilities, but we are working on the P2X and P2Y subtype of receptors. So there are the few drugs which are in the market. However, there are several. There are like only P2Y12 antagonists. They are in the market, and the P2Y2 agonist and is for the dry eye disease. However, there are several companies, they are trying to develop the selective antagonist of these receptors. So they have these receptors are antagonized by the different purinergic signaling molecules like ATP, ADP, UDP, and so on. So they have different outcomes. So that's why this is the structure of the P2Y receptor. So it is 7 transmembrane domain, and nitrogen amino is the outside, and the carboxy is the inside of the cell. So these are the different physiological roles in the body. And the purpose to develop the antagonist is to, it has the therapeutic outcome, like it relieves pain, antithrombotic effect, digestive cancer, epilepsy, and so on. So all of the receptors, they are unique and innovative in the sense that new pathways of the disease can be triggered out. So these are what we normally do. There are very less compounds available in the nature who are the antagonist or the agonist of the receptors, because they have very low activity. So that's why we mainly focus on the synthetic molecules. So we synthesize the compounds based on the knowledge from the literature as well as from the computational studies. We find we make the homology models, and then we screen the compounds based on the affinities. Then we compare with the literature, and then we design our own compounds. Similarly, we have the plasmid, then we transform into the like E. coli, then express in the mammalian cell lines, and then we perform the assays. So these are the receptors which we express in our lab. So these are their expression systems. And we have like without non-transfected, and then we have P2X, P2X2, P2X4, and P2X7 different receptors. So these are the already reported antagonists. So what we do, we took the literature and we search for the reported molecule, and then we try to incorporate our own different substitutions in the molecule, and then we find. So if we, from the literature, we see thiourea compounds and some sulfonamides and amide linkages, which are very important to be active for the molecule. So that's based on the literature knowledge, we design our own molecule, keeping all the ligands in the same place. based on our computational knowledge. So we synthesize different molecules of indomethacin derivatives, thiourea. So these thioureas, they bind inside the active site of the receptor. So that's why we put the thiourea inside the indomethacin. So we synthesize the different derivatives and then we analyze against the different receptors. So one of the compound for F with the two bromine items, they are, it is one of the most potent, the rest are relatively less potent. Similarly, we have another molecule which we synthesized, and we must like benzamidazole based sulfonyl urea compounds. So this is another scaffold which seems to be quite active. So we synthesize its derivatives, and then these are the different derivatives which we synthesized. So because the receptors, they are based on the cell membrane, so it is important to see their toxicity. If they are cytotoxic to the cell, so these are not worthwhile to be tested. So most of the compounds, they were quite safe. They have were less toxic on the cell lines which were used for the expression of the receptors. So here we have like P2Y1 receptor, it was quite active, the compound. And they are mainly with the chloro group and with the bromine group. So with the halogenated groups, they are more active. So these are the different selectivity between the different compounds. So with the nitro group, it was less active. Similarly, with the 2-chloro or 2-bromo, they were more active. So how the activity was increasing and how it was decreasing by taking the different substitutions at the different positions. So these are the structure-activity relationships. Similarly, we investigated several compounds on the inotropic receptors. So these are the P2X receptors. So they are different than the GPCRs. They are the 7 transmembrane. However, they have the 2 membranes, transmembrane 1 and 2. So they have also like different physiological roles in the body. So if we have the antagonists, they can be used as a brain disorders against the depression, inflammatory bowel disease, hypertension, and so on. So they are expressed in the different parts of the body. So here in the slide, you can see on the right side, these are the compounds reported by the different pharmaceutical companies, and based on this previous knowledge, we incorporated the different substitutions along with the admantane, and we again synthesize the admantane-based thiourea compounds. And similarly, we have another molecule using this, like we have the admantane-based thiourea and another molecule which is a pepronolic acid derivatives. So here in the slide, we have seen the functional group which were important, and we incorporated into the single molecule, and then we found and it was observed that these molecules were more potent than the reported compounds because we incorporated several active moieties in the single molecule. So these were compounds were also quite safe at the cell lines, so they were not cytotoxic. And these are the results which shows that most of the compounds, they were selective at one of the receptor, but they were not active at the other. The main the scaffold which were almost inactive, but when we incorporated different different molecules, then it was more active, and some of the compounds were selective. So these are the dose response curves of the different molecules, and we compared them with the already reported non-standard antagonist. So these are, we use the different acids, like this is a ethidium bromide acid and calcium flux acids. So various acids was employed to confirm that these compounds were active. So these are the different, another type of compounds with carboxylic acid thiourea. And similarly, when we study their binding affinity with the compound with the computational method, so there are 2 binding sites. One is the ATP binding site, and another is the allosteric binding site. So our compounds were mostly binding at the allosteric binding sites. So all classes of the compounds, they were binding at the allosteric binding site of the receptor. So this is pepronolic acid based thiourea compounds. We have the single crystal, which confirm the structure along with the NMR and other studies. So they were also quite safe at the normal cell lines and the receptor expressing the cell lines. So they were again tested, and again with the dibromo fluoro compounds, they were most potent. So with the halogenated compounds were active and the non-halogenated was less active. So we performed the calcium assays and compared the different selectivity with the different receptors. So these are the different, this is the structure activity relationship, which shows which part of the molecule is important and which functional group is important for the activity. So these are the how we synthesize the compounds. We incorporated different active compounds in the main scaffold, and then we find out they were more potent. So these are the different derivatives which were compared with the already reported patents from the different companies. So then we also performed the in vivo studies with the compounds, and we used the fluoxetine, and we found these compounds were like they have the antidepressant activity in the different animal models in vivo studies. So these are the different in vivo studies models. So we have the receptors, and then the cell surface we have the enzymes. So our target, we try to develop the antagonist, and similarly we also develop the inhibitor of the enzymes, because they are playing together the same role. So we have different enzymes like NTPases, NPPs, acto-5 nucleotidase. All of the enzymes, they dephosphorylate the adenosine triphosphate or diphosphate or adenosine monophosphate to the respective phosphates as well as the nucleosides. So we again find some of the compounds which were quite potent, and these values are in the micromoles. So we have also synthesized sulfonyl hydrazones. So these are the actonucleotidase inhibitors with the quite in the lower micromolar range, and it shows the binding affinity inside the active site, and it forms the hydrogen bonds with the metal in the active site of the enzymes. So these are the again thionopyrimidine compounds which are quite potent inhibitor, and these compounds are also not very toxic. They are very closely related to the nucleotides, other nucleosides. So these are the different molecules which we synthesized, and also we find their affinity in the computational studies. So this is our group and collaborations. So thank you very much, all of you, for listening after the lunch.
Thank you, Professor Jamshed Iqbal. You are well on time. Any question from Professor Jamshed Iqbal? Okay, sir, please.
Thank you very much, Doctor, for your information. And I'd like to know apoptotic body. I'd like to know that how it works, because in addition, it's a program. Can you throw some light on this principle, how it works, apoptotic body?
Actually, we are working on the receptors. We take the plasmid, like the genes, and express in the cells, and then we see the agonist or the antagonist. The pro-apoptotic antibodies, like these are the caspases and some other mechanisms for the anti-cancer, which we are not working on this topic. We just we are focusing on the purinergic signaling and the enzymes and the receptors related to the purinergic signaling. So we develop the computational methods, and we express our own receptors, and then we see the agonist or the antagonist effect using the calcium assays, functional assays. So it is a little bit different from the pro-apoptotic bodies and how they work, like P53 genes or some caspases, caspase 3, 9, or 7. So we have not investigated these compounds with respect to the cancer mechanisms. Rather, we just see their cytotoxicity, like if these compounds are not cytotoxic, mean we take the whole cell and we treat with the compounds. And if the cells are not dead, then that means the cells are safe. So we can make the functional assays. But if the cells, they are killed, or there is toxicity to the cells, then we cannot perform the assay because the receptors or the enzymes, they are on the cell membranes. So it is our main goal. We also perform some fluorescence microscopy to observe that cell surface is intact. So we are not focusing on the cytotoxic or anti-cancer compounds. We want to have our compounds are safe at the cells so that we can perform the functional assays.
Thank you, Professor Jamshed. Thank you so much. Okay, so please, Professor Jamshed, please. Dr. Ati has one question.
Sir, I just wanted to ask the plasma that you have transformed in the cell line. So you have expressed the protein and purified it, or you treated the cell having the plasmid with the compound?
So actually, we take the plasmid and it expresses the receptor, and the receptor is embedded in the cell membrane, like 7 transmembranes. So these are inside the cell. If we remove the receptor from the cells, it will not be active. And it is very difficult to remove because there is only one crystallographic structure available. Mostly there are homology models or with the electron microscope. So we perform the assays on the expressed proteins within the cell, on the cell membrane.
Thank you, sir.
Thank you.
Okay, thank you, Professor Jamshed. Our next speaker is Dr. Ulker Mamadova from Baku Institute of Botany, Mesra. The topic of our talk is phytochemical composition of species belonging to the genus Valeriana, distributed in the flora of Azerbaijan. So, please, Professor Ulker.
Hamınızı salamlayıram. Sormalarıma soruşacağım. Ben Azərbaycan dilində çıxışımı edeceğim. Ancaq slaytlarda ingilis dilində tərcümə olunub və mən onları sizə təqdim edirəm vaxtınızı çox almamaq üçün.
I would like to greet every one of you. And my speech will be delivered in Azerbaijan language, but the slides will be in English, so I will not take too much time.
Piyotu kimlər fəsilətinə daxil olan bəzi faydalı növlərin ehtiyatı və biokimyəvi əlamətlərinin analizi.
My speech will be about some species of belongs to Valerianaceae and their beneficial properties.
Azərbaycan florası növ tərkibinə görə zəngin olması ilə fərqlənir. Bioloji faal maddələr canlı həyatın vacib elementlərindən biridir. Ona görə də bu maddələrin öyrənilməsi, biologiya və eləcə də tibb elminin qarşısında duran əsas vəzifələrdən olub, müxtəlif təbii kimyəvi preparatların, o cümlədən dərman preparatlarının hazırlanmasına xidmət edir.
Azerbaijan flora is one of the richest flora in the world, and obtaining natural products, natural substances from the medicinal plants is one of the main goals, and one of the main goals of biology and medicine. And also, based on these, developing the medicinal products is
please, bit loud.
Bioloji faal maddələrin tükənməz mənbəyi olan bitkilərin axtarışı, onların ehtiyatlarını və kimyəvi tərkiblərinin müəyyən edilməsi, bu bitkilərin ehtiyatlarının əhalinin sağlamlığının qorunması və xalq təsərrüfatının ehtiyatları üçün istifadə imkanlarının işlənib hazırlanması, müasir dövrdə bitki-şünaslıq, tibb və sairə elmin aktual istiqamətlərindəndir.
Learning the resources of the plants and also obtaining the medicinal substances from them is one of the main goals of the current studies in terms of providing also the public health care system.
Məlumdur ki, bitkilərin faydalı xüsusiyyətləri əsasən onların kimyəvi tərkibi ilə bağlıdır. Azərbaycan florasında yayılmış valeriana cinsi növləri seskvitetin laktonlarla zəngin olur, istər praktik, istərsə də nəzəri cəhətdən böyük əhəmiyyət kəsb edir. Məhz buna görə də dünyanın bir çox laboratoriyaları öz tədqiqat işlərini seskvitetin laktonların öyrənilməsinə yönəltmişlər.
It's known that the beneficial effects of the plants is due to the chemical substances that are in them. And one of the species that we have in the research is the valeriana, belongs to valerianaceae. And the main components were the sesquiterpene lactones. And it's known that world laboratories are also studying sesquiterpene lactones in terms of their beneficial effects.
Bu birləşmələr qrupu geniş farmakoloji təsir spektrinə malikdirlər. Sintez yolu ilə alınmış birləşmələrdən fərqli olaraq az toksik olması və əlavə effektlər yaratmamasıdır. Pişiyotur cinsi növlərinin köklərindən alınan preparatlar müasir təbabətdə və xalq təbabətində, müxtəlif sinir sistemi xəstəliklərində, ürək-damar sistemi pozğunluqlarında, yuxunu tənzimlənmələrində, ürək fəaliyyətini normallaşdıran, eləcə də epilepsiyada işlədilir.
The medicinal substances that are obtained from the valeriana, have been utilized in some of the diseases, including epilepsy and cardiovascular systems and nervous systems.
Pişiyotu cinsi növlərinin dəmləməsi, sulu və spirtli tinkturasından, quru və qatı ekstraktlarından elmi təbabətdə geniş istifadə olunur. Bundan başqa, ürək xəstəliklərində istifadə olunan kompleks dərmanların tərkibinə də daxil edilmişdir.
The extracts and infusions obtained from the valeriana species have been utilized in modern medical systems, and also they have been used for against cardiac diseases.
Dərman vasitələrinin ümumi sayının təxminən 40% bitki mənşəli preparatlardır. Belə dərmanların istehsalı üçün Azərbaycan Respublikasında ən çox geniş yayılmış dərman bitkilərindən biri də dərman pişik otusudur.
The medicinal plants are one of the important of the pharmaceutical system, and one of the medicinal plants that we are studied is valeriana officinalis.
Bu bitkinin tərkibində efir yağları, valerian və hidrovalerian turşuları, üzvü turşular, alkaloidlər və bir çox digər qiymətli bioloji faal maddələr mövcuddur.
Organic acids, essential oils, and also alkaloids have been in this plant, valeriana officinalis.
Valeriana erotika növünün yerüstü hissələrinin sulu və etanollu ekstraktlarının bioloji faal maddələrinin analizi zamanı alkaloidlər, taninlər, kumarinlər, saponinlər, polifenollar və sairə usuzu yağlar aşkar edilmişdir. Antioksidant və antiproliferativ təsiri onun şiş əleyhinə istifadəsinə potensialını ortaya çıxarmışdır.
During our research on the aerial parts of valeriana erotica, we got the polyphenols and alkaloids, and their antioxidant activity and proliferative activity have, due to them, we have suggested for its use against cancer.
Pişiyotular fəsilləsinin pişiyotu cinsi növlərinin də bioloji faal maddələrinin çox olması aparılan eksperimental tədqiqatların nəticəsi ilə sübut edilir ki, valeriana erotika növünün köklərindən və yerüstü hissələrindən ilk dəfə olaraq bitki için yeni siz gibi tezden laktonlar alınmıştır.
In our research, first time we have obtained the sesquiterpene lactose from the roots and the aerial parts of this Valeriana erotica plant.
Bu cinsin növlərindən alınan biyoloji faal maddələr kardiotonik, iltihab əleyhinə, spazmolitik, antibakteriyal vasitələrdir.
Cardiotonic, antibacterial, and antispasmodically. These substances have been used against used as antispasmodic antioxidant agent.
Professor Səyyərə İbadullayeva və Ramiz Ələşbarov pişiyotu cinslərinin növlərinin köklərində 0,2% efir yağının olmasını və efir yağının əsas komponentlərinin pinen, borneol, kamfiol, izovalerian turşuları və bundan əlavə valerian turşusunun xatin alkaloidi, valerian glikozidi, aşı maddələri, üzvü turşular, şəkər tapılmasını qeyd etmişlər.
Professor Batulaiha and Alexander have conducted the research on this plant, and they have obtained 0.2% essential oils, and the ingredients of these essential oils were pinene, borneol, isovalerianic acid, and tannins and sugars.
Valeriana alliarifolia növünün kökümsovlarının tərkibində 0,52% efir yağı vardır ki, onun da miqdarı ekoloji faktorun təsirindən bitdiyi və ya becərildiyi şəraitdən asılı olaraq dəyişilməsi qeyd olunur. Efir yağının tərkibində borneol valerian efiri, izovalerian turşusu, mirfenol, izovalerian turşusunun efiri şəklində kamfen, pinen, terpenoidi, limonin, desgliterpen, qarışqa, sirkə və yağ turşularının borneol efiri ilə xarakterizə olunması aşkar olunmuşdur.
Another species is Valeriana erifolia, and during the research they have obtained 0.2% essential oils also, and it included borneol, isovalerianic acid, kamfen, pinene, limonin, and some of this is lactose.
Uzunçdə pişiyotu növlərinin fitokimyəvi tərkibi ilk dəfə tərəfimizdən öyrənilmişdir. Hər bitkinin həm yerüstü hissəsi, həm də yeraltı hissələri toplanmış və analiz üçün hazırlanmışdır.
Both the underground and above ground parts of two Valerian species have been studied first time by us, and its phytochemical components have been studied.
Biyoloji fəal maddələrin alınması iki istiqamətdə aparılmışdır. Belə ki, birinci istiqamət bitkinin kökü və çiçəklərindən biyoloji maddələr cəminin alınması üçün ekstraksiya üsulundan istifadə edərək ekstraktiv maddələr cəmi alınmışdır.
Two different paths we have followed. First one is getting the extracts from the roots and flowers.
Bu maddələr cəmi sütunlu xromatografiya üsulu ilə üzvü həlledicilərlə izolasiya edilərək fraksiyalara ayrılmışdır.
After that, we have conducted chromatography by using some of the organic solvents in order to get the fractions.
İkinci istiqamət isə alınmış nümunələr qaz xromatomat spektrometriya metodu vasitəsilə analiz edilmişdir.
The second direction, we have used mass chromatography, mass spectrometry methods in order to reveal and identify the components.
Bundan başqa sütunlu xromatografiya metodu ilə alınmış maddələrin fərdiliyini sulfiyol lövhələrində nazik təbəqəli xromatografiya metodundan istifadə etməklə təyin etmişdik.
Conducted the chromatography in order to identify the chemical substances.
Tədqiqat obyekti olaraq Valeriana erotica növünün xammalı çiçəklənmə fazasında da yığılmış və tədarüz edilmiş, xırda doğranmış və müvafiq qaydada qurudulmuşdur.
As an object, research object, we have used the roots of the plants. It has collected during the blossoming period of the plant. The roots have been chopped down and dried and processed for the upcoming
İlk defa olarak uzun çiçek pişiyotu növünün köklərindən izoalantalakton çiçəkleme fazasında, gövdəsindən və çiçəklərindən isə axilin hər iki növdən isə terpenoid və aromatik birləşmələrə ait komponentlər əldə olunmuşdur.
For the first time, we have isolated the isoallantalactone and ethylene from its roots, ethylene and another aromatic compounds we have identified.
Bundan başqa tədqiqat zamanı ikinci növ sarımsaq yarpaq pişiyotunun ədəbiyyat məlumatlarından fərqli olaraq başqa komponentlər də tərəfimizdən alınmışdır. Bu da fikrimizlə onunla izah olunur ki, ali bitkilərin kimyəvi tərkibi bir çox hallarda, xüsusən də müxtəlif iqlim və torpaq şəraitində bitkilərdə gedən fizioloji biokimyəvi proseslərin istiqamətini dəyişərək xeyli dəyişikliyə məruz qalır və bu zaman növ üçün xas olan kimyəvi birləşmələrlə yanaşı yeni maddələrin də aşkarlanmasına səbəb olur.
During our research on valeriana plants, as a part of higher plants, we have revealed that due to climate and other physiological factors, the chemical substances and their quantity, and also their number differs between the species.
Uzun çiçek pişiyotu növünün çiçəklərinin fitokimyəvi tərkibi qaz xromatoma spektrometriya metodundan istifadə edilərək tədqiq edilmişdir. Bitkinin çiçəklərindən alınmış maddələr cəmində müxtəlif qruplardan olan komponentlər cədvəlimizdə də verilmişdir, müəyyən edilmişdir.
And another species of valeriana genus, we have conducted again GCMS analysis on its flowers, and the component lists have been shown in the slides.
Bu cədvəldə isə uzun çiçək pişiyotu növünün çiçəklərindən aşkar olunan heksadekan tosusunun metil efiridir.
In the slides, we have shown the hexadecanoic acid, methyl ester of hexadecanoic acid, which we have isolated from the flowers of one of the valeriana species.
Burada isə uzun çiçək pişiyotu növünün çiçəklərində aşkar olunan bitkiklik monoterpenlər, naftalan, oktal hidrotrimetil trans spektrometriya edilmiş və şəkli burada qeyd edilmişdir.
In this slide, you have seen the cyclic monoterpenes which have been found from the flowers of long-flowered valeriana species.
Valeriana erotica növünün köklərinin tərkibində qaz xromatografiya kütlə spektrometriya üsulu ilə maddələrin identifikasiyasının nəticələri bu cədvəldə də öz əksini tapmışdır.
In this table, we have indicated the results of the GCMS components of the roots of the valeriana erotica.
Tədqiqat obyekti olaraq götürülən valeriana alliarifolia pişiyotu növü tərəfimizdən yığılaraq qurudulmuş və çiçəklənmə fazasında yığılmış və qurudulmuşdur və xırda doğranmış asetonda ekstraksiya edərək alınmış ekstraktiv maddələr cəmindən istifadə edilmişdir.
To the species of valeriana alliarifolia, we have collected its underground and above ground parts and made an acetonic extract and then conducted the other analysis.
Valeriana alliarifolia növünün kökləri və yerüstü hissələri asetonda ekstraksiya olunmuş, alınan bioloji maddələr cəmi sütun xromatografiya üsulu ilə fraksiyalara ayrılmış və komponent tərkibləri qaz xromatografiya kütlə spektrometriya metodundan istifadə edərək təyin edilmişdir və nəticədə 5 komponent identifikasiya edilmişdir.
From the collected roots and aerial parts of valeriana species, the biologically active substances have been revealed by using column chromatography first and then gas chromatography mass spectrometry method. And at the end, we have revealed 5 different components.
Bu şəkildə isə sarımsaq yarpaq pişiyotunun qaz xromatoma spektrometri metodu ilə tədqiqinin nəticələri göstərilmişdir.
In this picture, you have seen the GCMS results of the another species of Valeriana.
Beləliklə, ilk dəfə olaraq Azərbaycan florasında yayılan Valeriana alliarifolia növünün yerüstü hissəsində 5 maddə aşkar edilmişdir.
For the first time, the Valeriana alliarifolia species, the aerial parts of it, we have revealed 5 different substances.
Qaz spektroskopiya metodlarından istifadə edərək maddələrin identifikasiya edilməsi, uzun çiçək pişiyotu bitkisinin köklərindən və çiçəklərindən dəmir 8 komponent identifikasiya edilmişdir. İlk dəfə olaraq uzun çiçək pişiyotu bitkisinin çiçəkləmə fazasında toplanan bitki köklərindən bitki üçün yeni maddə izoalantalakton, çiçəklənmə fazasında çiçəklərindən bitki üçün isə yeni maddə axildən seskili terpon laktonları alınmışdır.
A total of 8 components have been identified from the roots and flowers of the long-flowered valerian plant. For the first time, new substance for plants, isolantalacton, was obtained from the roots of the long-flowered valerian plant collected during the flowering phase. The quality and composition of the chemical components of the roots and flowers of the species were studied using the GCMS method.
Beləliklə, apardığımız eksperimental tədqiqatlar nəticəsində tədqiq etmişdir ki, Valeriana erotika növünün yüksək antioksidant fəallığa malik, praktiki cəhətdən qiymətli olan izoalantalakton, axildən seskili terpon laktonların miqdarının çox olması və eyni zamanda ehtiyatlarının az olmasını nəzərə alaraq toxum vasitəsilə çoxaltılması nəzərə tutulmuşdur.
And as a result, we have revealed the isoalantalacton and acetylene 6-terpene lactones. And due to their high antioxidant activity and practical availability, at the same time, they have more results. We have suggested their artificial reproduction by seeding.
Diqqətinizə görə təşəkkür edirəm.
Thanks for your attention.
Thank you, Professor Olkar, for such a nice presentation. Any question from Professor Olkar? So, you have question? Okay, thank you so much. So our last speaker, Ms. Laila Ahmadova. She's also my student, also working in my course supervision. And also representing Institute of Botany. Our topic is useful plants of miniature reservoirs area. This area is basically wetland, about 17 kilometers away from Baku. And it is on both sides of the river. So it is very touristic place near Baku. So please, Laila.
Salam, ladies and gentlemen. My topic is useful plants of the miniature reservoir area, and I will keep my speech short. The miniature reservoir is the largest artificial lake in the South Caucasus, hosts a diverse flora with over 600 species of vascular plants. Many of these species are valuable both ecologically and economically, serving as sources of food, medicine, and raw materials. Among these medicine plants can be given as examples. Plantago major accelerates wound healing, has anti-inflammatory properties, and is used in respiratory diseases. Achillea minifolium has antiseptic and hemostatic effects, widely applied in both medicinal practice and folk medicine. Hyperseum perforatum used for nervous system disorders, gastrointestinal problems, and skin diseases. Artemisia
absinthium improves digestive functions, and its essential oil are used as raw material in the pharmaceutical industry. Edible and food plants, allium, vinyl, and other wild onion garlic species rich in vitamins consumed fresh or as a spice. Rubex acetosa, recognized as a source of vitamin C, included in the human diet, has been particularly important in preventing spring hypervitaminosis. Urtica diosa, used as both food supplement and medicinal plant, rich in protein, therefore of high nutritional value. Forage plants, Piscuta arundinata and Poa pratensis form the forage base for livestock, dominating pastures due to high adaptability. Mediaco sativa, rich in protein, one of the main forage plants for animal husbandry. Industrial and economic plants, tamarisk species play an important role in soil salinity control. The creation of windbreak belts and the stabilization of landscapes. Populis alga, a fast-growing tree species valued as a source of timber and fuel. Artemisia species, industrially important for essential oils and biologically active compounds. Ecological and household use plants. Pamicus australis acts as a natural filter in water purification and prevents soil erosion. Also used in roofing, basket, bearing, and household crafts. Tufa angustoplia serves as a bioforming species with seeds that can be used as thermal insulation material. Among the list plants, only some species of the genus Allium are included in the Red Book of the Republic of Azerbaijan. The useful plants of the Mingechiri Reservoir area are important resources from both ecologically and economic perspectives. Their use in medicine, food, forage, and industry is invaluable for human health, agriculture, and daily life. The conservation and sustainable use of these plants are essential for maintaining regional biodiversity and ensuring ecosystem stability. I'm glad to be here. Thank you so much.
Thank you, Leila, for an overview of flora of wetlands. Any question from Leila? Okay, thank you so much, Leila. So the forum is open for any kind of question or suggestion regarding medicinal, aromatic, and cosmetic plants.
Okay, ladies and gentlemen, with this we come to the session 2, technical session of this workshop, which is the last one. And now we will be moving on to the closing ceremony of this event. And for the distribution of certificates, I would request Professor Dr. Sayara and Professor Dr. Mudassir Asrar to please award certificates to our resource persons who took out their valuable time and to share and impart their knowledge and contributions. Okay. यहीं पे मैडम नो जी जी मैडम हाँ Okay, so first, I would request Professor Dr. Dilzara to come and receive her certificate. Professor Dr. Raza Shah, sir, thank you very much for coming. Lectures पे जाना है फोटोग्राफर बोल दो कब Professor Dr. Atiyatul Mahaf. Professor Dr. Ehsan Adad. Dr. Jamshed Iqbal. Do we have Dr. Jamshed Iqbal here? No. So, Professor Dr. Nigar Mursal. Dr. Hilal. Dr. Ayman. Dr. Ayman. Dr. Ulkar. Miss Laila. Mr. Ibrahim Jafar. Okay. I would now request all our speakers to please come in the front for a group photo of all the speakers with Professor Dr. Seara and Professor Dr. Mudassir Asrar. Madam, please, here. Dr. Hilal, Dr. Laila, Ibrahim, please come. Dr. Raza, Dr. Asana is here, Dr. Atiya. Please give them a big round of applause for their effort and for their contribution. Thank you very much. Now I would request Dr. Seara to please say a few words, madam. You can be there and the mic is there. Yes. Emil, you can come here.
Buraya mı bakalım? Çok şey yazmışım. hörmətli təşkilatçılar, dəyərli həmkarlar, əziz dostlar,
Dear esteemed researchers, colleagues, and friends,
seminarın bugünkü müzakirələri və təqdimatları mənə dərindən təəssürat bağışladı.
I have been influenced, extremely influenced by the today's presentations,
Xüsusilə Karachi Universitetinin həmkarları Professor Atiya və Professor hər ikisinin çıxışından yəni biz özümüz üçün də yeni bir yollar gələcək üçün bir vizyon hazırladıq.
I have been also influenced by the speeches delivered by Professor Atya and Professor Razesha, which were the participants of the University of Karachi, and we will take their
Və digər çıxışların da eləcə də çıxışları.
also this is slide for all the speeches delivered today here.
Burada əldə olunan biliklər, ideyalar, elə bilirəm ki, və eləyirəm ki, həm bu təcrübələr Pakistan gənc mütəxəssislərinə və Azərbaycan mütəxəssislərinə gələcəkdə yeni elmi əməkdaşlıqlar aparmasına təkan verəcək.
Ideas and experiments, experiences that have been we have been witnessed today, but they will be a foundation and inspiration for the future Pakistani and Azerbaijani youth development.
Digər tərəfdən bizim Pakistanlı dostlarımız, həmkarlarımız var burada. Onlara da təşəkkür edirəm. İclasın gedişində dəstək olduğu üçün Məhəmməd Zəfərə və xanım Əsiyə xanıma.
On the other hand, I would also like to especially thank to Professor Atya and Professor Mohammed Zafar for their help and support.
Və ümid edirəm ki, bundan sonra qarşılıqlı əməkdaşlığımız daha da geniş olacaqdır.
I hope that our collaborative cooperation will widen from now on.
Və bugün burada bir çox universitetlərin gənc xanımları mənə yaxınlaşdı, gələcəkdə tədqiqatlar aparmağa, müştərək tədqiqatlar.
For future joint experiments, today I have been questioned by some of the young female students of other countries.
Yəqin ki, yaxın gələcəkdə mən onlara dönəcəyəm və onlara fikirlərimi yazacağam və Azərbaycana dəvət edəcəyəm.
In the near future, probably I will get back to them. I will give them my feedback and I will also invite them to Azerbaijan.
Və buradan xüsusi təşəkkür eləmək istəyirəm Professor Məhəmməd İqbal Çövdariyə. Çünki o bu workshopun təşkilində çox isti yanaşdı və bizi dəvət etdi. Ona görə ona çox təşəkkür edirəm.
My very special thanks go to, of course, General Coordinator of the conflict, Professor Dr. Muhammad Iqbal Chaudhary, because he has very warm welcomed us and hosted us here.
Və deyərdim ki, bu workshopun keçirilməsində ən çox xidmətləri olan xanım Xazimaya çox təşəkkürümü bildirirəm.
Another special thanks goes to Madam Kazima for organization of the...
O nəinki bu workshopun keçirilməsində, hətta bizim qarşılamamızda, səhər, gün orta, axşamda yeməyimizdə belə vaxt sərf edir və bu barədə düşünür. Ona görə çox təşəkkür edirəm Xazimaya.
She also takes too much time for organizing the breakfast, lunch and dinner events. And also she have welcomed us, brought us from airport to here.
Əsas təşəkkürlərimdən biri də mənim Professor Doktor Atiya xanımdır.
Another special thanks go to Professor Dr. Atya.
Biz Atiya'dan bir ildən çoxdur tanışıq. Amma elə bilirəm ki, mən bütün həyatım boyu Atiyanı tanımışım.
It's just more than 1 year that we have been introduced, but it looks like that we have introduced ever since.
Bütün iştirakçılara teşekkür ederim. Bütün çıkışçılara iki kat teşekkür ederim. Çok sağ olun, var olun. Allah emanetinde olun.
I thank everyone, to all participants, all of those who delivered speech today. Thanks for your attention.
Allah emanetinde olun demedi.
May God bless all of you.
Allah. Sağ olun, var olun.
Okay, ladies and gentlemen, thank you very much. We come to end of our workshop. All the participants, they have been provided their certificate of attendance. Thank you very much for joining. And for the online participants that we have, we will be providing them any certificate soon, inshallah. And also there is an announcement that the AI certificate course that we did run for more than 20 days almost. The certificates will be given to you on the e-certificates soon. So thank you very much, and thank you for your patience and for your valuable time here. The tea is outside. Please join us.